So good evening everyone, good evening, welcome to the Deeksha Vedantu 9th and 10th. ನಾನು ನಿಮ್ಮ ಮಾಸ್ಟರ್ ಟೀಚರ್ ಬಸವರಾಜ್. ಸೊ ಇವತ್ತಿನ ಸೀರೀಸ್ ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ಸೆಷನ್ ವಿಲ್ ಬಿ ಫ್ರಮ್ NCERT line by line. That is, I'll be teaching you the entire chapter. Our entire chapter is how do organisms reproduce. How do organisms reproduce? A complete chapter ನಾನು ಕಂಪ್ಲೀಟ್ ಆಗಿ NCERT ಲೈನ್, ಎವ್ರಿ ಸಿಂಗಲ್ ಲೈನ್ ಆಫ್ NCERT ನಿಮಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ಸೊ ಈ ಚಾಪ್ಟರ್ ನಾವು ಕಂಪ್ಲೀಟ್ ಆಗಿ ಮುಂಚೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ, ಫುಲ್ 3 ಪ್ರಾಪರ್ ವಿಡಿಯೋಸ್ ಆಗಿದೆ. ಬಟ್ ಇಟ್ ಇಸ್ ಆಲ್ವೇಸ್ ಇಂಪಾರ್ಟೆಂಟ್. ಆ NCERT ಲೈನ್ಸ್ ಇದೆಯಲ್ಲ, ದೀಸ್ NCERT ಲೈನ್ಸ್ ಬಿಕಮ್ಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ಯು ಟು ಸ್ಟಡಿ. ಸೊ ಇನ್ ದಿಸ್ ವಿಡಿಯೋ, in this session, I'll be making sure every line of NCERT, every single word of NCERT is covered and you'll be able to understand. So ಈ ಹ್ಯಾಬಿಟ್ ಇದೆಯಲ್ಲ, ರೀಡಿಂಗ್ ಆಫ್ NCERT ಲೈನ್ಸ್, ಈ ಯೂಶಲಿ ನಾವು ಕ್ಲಾಸ್ 12 ಮತ್ತು ಕ್ಲಾಸ್ 11th 12th ವರೆಗೆ ಹೇಳ್ತೀವಿ. ಬಿಕಾಸ್ ನೀಟ್ಗೋಸ್ಕರ ಎವ್ರಿ ಸಿಂಗಲ್ ಆಫ್ ಲೈನ್ ಆಫ್ NCERT ಇಂಪಾರ್ಟೆಂಟ್, ಬಿಕಾಸ್ ಎವ್ರಿ ಸಿಂಗಲ್ ಲೈನ್ ಆಫ್ NCERT ಒಂದು ಕ್ವೆಶನ್ ಬರ್ಬೋದು. ಬಟ್ ದಿಸ್ ಹ್ಯಾಬಿಟ್ ನೀಡ್ಸ್ ಟು ಬಿ ಇನ್ಕಲ್ಕೇಟೆಡ್ ಇವನ್ ಇನ್ ದ ಯಂಗರ್ ಕ್ಲಾಸಸ್. ದಟ್ ಇಸ್ ಕ್ಲಾಸ್ 9th ಆಗಿರಲಿ, ಕ್ಲಾಸ್ 10th ಆಗಿರಲಿ, ದಿಸ್ ಹ್ಯಾಬಿಟ್ ಆಫ್ ರೀಡಿಂಗ್ NCERT is extremely important. So before I start the session, I want all of you to hit the subscribe button. ಈ ಸಬ್ಸ್ಕ್ರೈಬ್ ಬಟನ್ ಕಾಣಿಸ್ತಿದೆಯಲ್ಲ, ಇದು ಎಲ್ಲರೂ ಸಬ್ಸ್ಕ್ರೈಬ್ ಬಟನ್ ಒಂದ್ಸರಿ ಜೋರಾಗಿ ಒತ್ಬಿಡಿ and I will start. So with that being said, ನಮ್ಮ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ಕೂಡ ಇದೆ. ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ದಟ್ ಇಸ್ ದೀಕ್ಷಾ ವಿಧಾಂತು 9th and 10th. ಈ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲ್ ನಾವು ಎಲ್ಲ ನೋಟ್ಸ್, ಎಲ್ಲ ಅಸೈನ್ಮೆಂಟ್ಸ್, ಎಲ್ಲ ನೋಟ್ಸ್ ಮತ್ತು ಡೈಲಿ ಅಪ್ಡೇಟ್ಸ್ ಈ ಚಾನಲ್ ನಾವು ಲೈವ್ ಆಗಿ ನಿಮಗೆ ಕೊಡ್ತಾ ಇರ್ತೀವಿ. ಸೊ ದ್ಯಾಟ್ ಇಸ್ ಇಟ್. ಲೆಟ್ಸ್ ನಾಟ್ ವೇಸ್ಟ್ any time anymore. Let's start a chapter, that is how do organisms reproduce. NCERT line by line, that is every line of NCERT, every single line of NCERT ನಾವು ಈ ಸೀರೀಸ್ ಅಲ್ಲಿ ಕವರ್ ಮಾಡ್ತೀವಿ. that is NCERT line by line for biology. So chapter number 7, one ನಿಮಿಷ, chapter number 7, right? Chapter number 7, how do organisms reproduce. So each chapter now we'll be studying about different types of reproduction. That is asexual reproduction ಕಲಿತೀವಿ ನಾವು, asexual reproduction ಕಲಿತೀವಿ. Then we'll be learning about what is different types of sexual methods, what are different types of asexual methods, different types we'll be learning about budding, spore formation, fragmentation, many different methods of different types of asexual reproduction. And under sexual reproduction we'll be learning about plant sexual reproduction as well as human sexual reproduction. So let's not waste any time, let's get into it. The first paragraph ನೋಡಿ, ಕರೆಕ್ಟ್ ನಾವು ಓದ್ತಾ ಇರ್ತೀನಿ, ಕೇಳಿಸ್ಕೊಂತ ಹೋಗ್ತಾ ಹೋಗಿ. ನೀವು ಸುಮ್ಮನೆ NCERT ಜೊತೆಗೆ ಲೈನ್ ಇಟ್ಕೊಳ್ಳಿ. ನಿಮ್ಮ NCERT ಓಪನ್ ಮಾಡಿ, ಎವ್ರಿ ಸಿಂಗಲ್ ಲೈನ್ ಐ ರೀಡ್ ಫ್ರಮ್ ಹಿಯರ್, ನೀವು ಕೂಡ ಆ ಲೈನ್ ನೀವು ಓದಬೇಕು. ಸೊ ಐ ವಿಲ್ ರೀಡ್ ಅ ಲೈನ್, ನೀವು ಸೇಮ್ ಲೈನ್ ಓದಿ. ಸೊ ಬಿಫೋರ್ ವಿ ಡಿಸ್ಕಸ್ ದ ಮೆಕ್ಯಾನಿಸಮ್ ಬೈ ವಿಚ್ ಆರ್ಗನಿಸಮ್ಸ್ ರೀಪ್ರೊಡ್ಯೂಸ್, ನಾವು the way organisms reproduce ಮಾಡುತ್ತೆ ಅದು ಕಲಿಯಕ್ಕಿಂತ ಮುಂಚೆ ನಾವೇನು ಮಾಡಬೇಕು, let us ask more about the basic question. Why do organisms reproduce? ಈ reproduction process ಇದೆಯಲ್ಲ, why is it even required? Will we be able to survive without reproduction? We will be able to survive. Like reproduction ಆಗಿಲ್ಲ ಅಂದ್ರೆ we'll be able to survive, but the continuity ಇದೆಯಲ್ಲ, ಆ ಕಂಟಿನ್ಯೂಟಿ ಆಫ್ ಆರ್ಗನಿಸಮ್ಸ್, ಕಂಟಿನ್ಯೂಟಿ ಆಫ್ ಸ್ಪೀಸೀಸ್ ಆನ್ ದ ಅರ್ತ್, ಆ ಕಂಟಿನ್ಯೂಟಿ ಆಫ್ ಸ್ಪೀಸೀಸ್ ಆಗಲ್ಲ ಅಷ್ಟೇ. ದಟ್ ಇಸ್ ದ ಮೇನ್ ಡಿಫರೆನ್ಸ್. ಸಿ ಆಫ್ಟರ್ ಆಲ್ reproduction is not necessary to maintain the life. ಹೇಳಿದ್ನಲ್ಲ, reproduction is not required to maintain the life, but to maintain the life on the earth, then it is required. So of an individual, unlike the essential life process such as nutrition, respiration, excretion is so important, because without these 3 processes a human or animal cannot survive. But without reproduction, an animal can survive without reproduction. So if you're able to hear me, right, if you're able to hear me and see me clearly, thumbs up in the chat if you're able to see me and hear me clearly. On the other hand, if an individual organism is going to create more individuals, if an individual organism is going to create more individuals, a lot of its energy will be spent in the process. That is, reproduction process ಇದೆಯಲ್ಲ, it is a lot of energy consuming process. When this process is taking place, a lot of energy is being consumed by the organism. So why should an individual organism waste energy on a purpose it does not need to stay alive? It would be interesting to discuss the possible answers in the classroom. So if it is not required, why survive ಆಗೋದಕ್ಕೆ ಈ reproduction process necessary ಇಲ್ಲ, then why is the reproduction process even happening? ಯಾಕೆ ಆಗ್ತಿದೆ process? The answer is very simple. The answer is the continuity of life. ಅಕಸ್ಮಾತ್ reproduction ಆಗ್ತಾ ಇಲ್ಲ ಅಂದ್ರೆ you will not see any organism around you. ನೀವು ರೋಡ್ ಹೋದ್ರೆ ನಾಯಿ ಕಾಣಿಸುತ್ತೆ, ಬಿಗ್ ಕಾಣಿಸುತ್ತೆ. If there was no reproduction, you would not see cats and dogs on the streets also. Whatever the answer to this question, it is obvious that we notice organisms as because they reproduce. Very important line. Just because reproduction is taking place, if reproduction is taking place, we are able to see the animals. We're able to see the animals around us. The elephant, panda. If there was no reproduction, we would not be able to see all these organisms. If there were to be only one non-reproducing members of a particular kind, it would be doubtful that we would have existed its existence. Notice existence. For example, ಇದು liger ಗೊತ್ತಾ? Liger, mule. These are the crossbreeds. Horse ಮತ್ತೆ donkey ಮತ್ತೆ mule ಅಂತ ಬರುತ್ತೆ crossbreed. But these mules are sterile in condition, that is they cannot reproduce. ಅದೇ reason ಗೋಸ್ಕರ you will not see a lot of mules in the nature. And liger is a cross between tiger and a lion. That is the same reason you do not see a lot of ligers in the environment. Only because of human intervention, humans crossbreed ಮಾಡಿದಕ್ಕೆ it's able to survive. But it is sterile in condition. It is not able to reproduce. So you cannot see in nature. Naturally, without human intervention, you will not be able to see all these animals such as tiger, liger and mule. Without human intervention, you cannot see. So if you have to see the organisms around you, ಬೆಕ್ಕು ನಾಯಿ ನೋಡ್ಬೇಕಂದ್ರೆ reproduction is extremely necessary. It is large number of organisms belonging to that single species that bring them us to notice. So lot of crows, lot of sparrows, lot of plants, lot of mango trees. So when there are a lot of mango trees, we will be able to identify in the nature. ಕೇವಲ ಒಂದೇ mango tree ಇದ್ರೆ world ಅಲ್ಲಿ, will you be able to notice this? Then there is going to be a very slim chance. There is a very slim chance that you will notice this mango tree. ಇಲ್ಲಾಂದ್ರೆ ನೋಟಿಸ್ ಮಾಡಕ್ಕೆ ಆಗಲ್ಲ ನೀವು mango tree ನ. So the only way you'll be able to notice the organisms is when there are a lot of them are present. If a lot of them are present, you'll be able to notice them. And then I'm sure if you're able, how do we know that two different individual organisms belong to the same species? That is similar characteristics irutte. Usually we say this because they look similar exactly to each other. How can we tell this cat and that cat belong? On German Shepherd nayi ಮತ್ತು golden retriever nayi, how do we tell them both are dogs? Because both of them walk on the four legs, both of them look alike, right? ಸ್ವಲ್ಪ difference ಇರುತ್ತೆ, but in the end of the day we can identify German Shepherd on doggo, golden retriever on doggo, Labrador dog on doggo ಅಂತ, because they look alike. Even though they might have little differences, but they still look alike. Organisms create a new individual that look very much similar to themselves. Thus, reproduction is a biological process in which organisms create a new individual. So reproduction is a biological process in which an organism create a new individual which looks similar to itself. So same to same ಕಾಣಿಸುತ್ತೆ. The new organism which is born, right? It is very similar to the parent. It is very similar or identical in some cases to the parent organism. Okay, so that was the before. ಈ chapter start ಆಗಿನ ಮುಂಚೆ all the information which you need to know before the starting of the chapter. Let's start the chapter. Do organisms create exact copies of themselves? ನಾನು ಹೇಳಿದೆ, they look similar, but do they create exact copies ಆಗುತ್ತಾ? Same to same, 100% doubt. Listen to it very carefully now. Organisms look similar because their body design are similar. If body designs are to be similar, the blueprint of these designs should be similar. ಅರ್ಥ ಏನಪ್ಪ ಇದು? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನೀವು ಕ್ವಾರ್ಟರ್ಸ್ ನೋಡಿದ್ದೀರಾ, ಇದು ಆರ್ಮಿ ಕ್ವಾರ್ಟರ್ಸ್ ಆಗಿರ್ಬೋದು, ಬ್ಯಾಂಕ್ ಕ್ವಾರ್ಟರ್ಸ್ ಆಗಿರ್ಬೋದು, ಈ ಅಪಾರ್ಟ್ಮೆಂಟ್ಸ್, ಈ ಎಲ್ಲ ಅಪಾರ್ಟ್ಮೆಂಟ್ಸು ಆಚೆ ಕಡೆಯಿಂದ ಒಂದೇ ಥರ ನೋಡೋಕೆ ಇರುತ್ತೆ. ವಿ ಕ್ಯಾನ್ ಸಿ ಫೈಂಡ್ ಆಲ್ ದೀಸ್ ಅಪಾರ್ಟ್ಮೆಂಟ್ಸ್ ಲುಕ್ ಅಲೈಕ್. ಯಾಕೆ ಲುಕ್ ಅಲೈಕ್? ಬಿಕಾಸ್ ಆ ಅಪಾರ್ಟ್ಮೆಂಟ್ ಕಟ್ಟಬೇಕಾದ್ರೆ ಆ ಬ್ಲೂ ಪ್ರಿಂಟ್ ಇತ್ತಲ್ಲ, ಅದು ಕಟ್ಟಬೇಕಾದ್ರೆ ಬ್ಲೂ ಪ್ರಿಂಟ್ ಸೇಮ್ ಆಗಿತ್ತು. ಸಿಮಿಲರ್ಲಿ ಇಫ್ ಅ ಹ್ಯೂಮನ್ ಅಂಡ್ ಅದರ್ ಹ್ಯೂಮನ್ ಲುಕ್ ಸಿಮಿಲರ್, ದಟ್ ಇನ್ ಎರಡು ಕಾಲು ನಡೀತೀವಿ, ಎರಡು ಕಣ್ಣಿದೆ, ಎರಡು ಕೈ ಇದೆ, ತಲೆ ಮೇಲೆ ಕೂದಿದೆ, ಎರಡು ಕಣ್ಣಿದೆ, ಒಂದು ಮೂಗಿದೆ. ಯಾಕೆ? ಬಿಕಾಸ್ ಆರ್ ಬ್ಲೂ ಪ್ರಿಂಟ್, ದಟ್ ಇಸ್ ಆರ್ DNA is similar. Thus, reproduction at its most basic level involves making copies of the blueprint of its body design. ಅರ್ಥ ಏನಂತಂದ್ರೆ, during the course of reproduction, during the process of reproduction, the main activity which is happening is copying of the blueprint. That is, DNA copy ಆಗ್ತಿದೆ. In class 9th, we learned that chromosome in the nucleus of cell contains information for inheritance of features from parent to the next generation in the form of deoxyribonucleic acid, that is DNA molecule. A DNA in the cell nucleus is the information source for making proteins. If the information is changed, different proteins will be made. Different proteins will eventually lead to the altered body design. Full paragraph ಏನು ಹೇಳ್ತೀವಿ ಅಂತಂದ್ರೆ, the information, the blueprint of the body is stored inside the DNA. So every single information, ಎರಡು ಕೈ, ಎರಡು ಕಣ್ಣು, ಒಂದು ಮೂಗು, ಒಂದು ಬಾಯಿ, ಈ ಮಾಡಬೇಕಿದ್ದ ಬ್ಲೂ ಪ್ರಿಂಟ್ ಇದೆಯಲ್ಲ, ಈ ಬಾಡಿ ಡಿಸೈನ್ ಬ್ಲೂ ಪ್ರಿಂಟು ಎಲ್ಲ DNA ಒಳಗಡೆ ಇದೆ. So if you able to copy the DNA, you're able to make an exact copy of the organism. And where is this DNA? DNA is present on the chromosome. Where is chromosome? Inside the nucleus. chromosome is present inside the nucleus. Nucleus illide. Nucleus is inside the cell ಒಳಗಡೆ ಇದೆ. Right? ಇಲ್ಲಿ ನೋಡಿ. We have learned that chromosome is on the nucleus of a cell containing information of inheritance. So all the inheritance of information is DNA ಒಳಗಡೆ ಇದೆ. So all the information of all the body design is everything is stored inside the DNA. So all the information is stored inside the DNA. Now if the information is changed, ಅರ್ಥ ಏನು? If the DNA is changed, ಬರ್ಕೊಳ್ಳಿ ಇಲ್ಲಿ ಸೈಡ್ ಅಲ್ಲಿ. If DNA ಇದು ಇಲ್ಲಿ ನೋಡಿ, ಒಂದು ನಿಮಿಷ ಬೇರೆ ಕಲರ್ ಹಾಕದ ಇಲ್ಲಿ. If the information change ಅಂದ್ರೇನು? ಒಂದೇ ನಿಮಿಷ. If DNA is changed, DNA is changed. One mutation ಆಗಿರಬಹುದು. How? Because of some mutation ಆಗಬಹುದು. Mutation ಅಂದ್ರೆ sudden inheritable change. ಇಲ್ಲ ಅಂದ್ರೆ during the reproduction, during sexual reproduction ಈ DNA ಚೇಂಜ್ ಆಗಬಹುದು. DNA ದ ಆಫ್ಸ್ಪ್ರಿಂಗ್ ಚೇಂಜ್ ಆಗುತ್ತೆ, ಪೇರೆಂಟಲ್ ಆಗಲ್ಲ, ನೆನಪಿರಲಿ. Right? Therefore, a basic event in reproduction is the creation of DNA copy. So the entire process of reproduction, the main design, the main thing which is happening, the main event which is happening is copying of the DNA, because the DNA has what? DNA ಒಳಗಡೆ ಏನಿದೆ? DNA ಒಳಗಡೆ the blueprint ಇದೆ. Blueprint of what? Blueprint of body design. So reproduction event ಅಲ್ಲಿ DNA copy ಆಗುತ್ತೆ, and ಈ DNA ಒಳಗಡೆ ಏನಿರುತ್ತೆ? Information ಇರುತ್ತೆ. So very important to note. And any change in the blueprint, ಈ ಬ್ಲೂಪ್ರಿಂಟ್ ಇದೆಲ್ಲ DNA ಏನೋ ಸ್ವಲ್ಪ ಚೇಂಜ್ ಆದರೂ ಕೂಡ ಡಿಫ್ರೆಂಟ್ ಪ್ರೋಟೀನ್ಸ್ ಬರುತ್ತೆ. ಡಿಫ್ರೆಂಟ್ ಪ್ರೋಟೀನ್ಸ್ ಬಂದರೆ ಡಿಫ್ರೆಂಟ್ ಬಾಡಿ ಡಿಸೈನ್ ಬರುತ್ತೆ. ಸೊ DNA ಅಲ್ಲಿ ಚೇಂಜ್ ಆದರೆ ಫುಲ್ ಬಾಡಿ ಡಿಸೈನಲ್ಲೂ ಕೂಡ ಏನಾದರೂ ಚೇಂಜ್ ಬಂದೇ ಬರುತ್ತೆ. Now, cells use chemical reactions to build copies of their DNA. Cell ಒಳಗಡೆ chemical reactions ಆಗುತ್ತೆ, different type of chemical reactions ಆಗುತ್ತೆ cell ಒಳಗಡೆ, that will bring about the chemical change. This creates 2 copies of the DNA in a reproduction cycle. Very important. Now this is giving you a hint. One hint ತೋರಿಸಿದೆ that this line is a hint. This line is a hint for semi-conservative method of DNA replication. I'll repeat again. This line, chemical reaction to build copies of DNA. This creates 2 copies of DNA in the reproducing cell. 2 copies of DNA. That is semi-conservative method of DNA replication. ನೀವು ಅದನ್ನ ಕ್ಲಾಸ್ 11th ಅಲ್ಲಿ ಕಲಿತೀರಾ. And they will need to separate it from each other. ಎರಡು DNA ಕಾಪಿ ಆಗುತ್ತೆ ನೋಡಿ. ಇಲ್ಲೊಂದು ಮಾಡೋದ, ಇದೆಲ್ಲ ಎರೇಸ್ ಮಾಡೋದ, ಕರೆಕ್ಟ್ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. ಸೊ ಒಂದು DNA ಇದೆ. This is your DNA. This is the DNA. ಈ DNA ಎರಡು ಪೀಸ್ ಆಗುತ್ತೆ. This will divide into 2 parts. We know DNA is double stranded. If the 2 strands are there, both strands are separated. Keeping 1 copy of DNA in the original. So ಇದು original cell ಆಗುತ್ತೆ. ಒಂದು ನಿಮಿಷ ಚಿಕ್ಕದಾಗಿ ಮಾಡೋದ. So this is your DNA. DNA will make 2 copies. ಈ 2 ಕಾಪಿಯಿಂದ 1 ಕಾಪಿ ಇದೆಯಲ್ಲ, ಬೇರೆ ಕಲರ್ ಯೂಸ್ ಮಾಡೋದು. ಈ ಎರಡರಲ್ಲಿ ಒಂದು ಒರಿಜಿನಲ್ ಇರುತ್ತೆ. ಈ ಒಂದು ಒರಿಜಿನಲ್ ರೆಪ್ಲಿಕೇಟ್ ಆಗ್ಬಿಟ್ಟು ಇನ್ನೊಂದು ಸಪ್ರೇಟ್ ಆಗುತ್ತೆ. This is duplicate one. This is the duplicate copy. However, keeping 1 copy of DNA in the original cell and simply pushing the other cell out would be not work. A cell ನ ಒಂದು DNA ದಲ್ಲಿ ಒಂದು ಕಾಪಿ ಮಾಡಿದಾಗ ಕಾಪಿ ಆಗುತ್ತಲ್ಲ, ಒಂದು ಒರಿಜಿನಲ್ ಒಂದು ಡೂಪ್ಲಿಕೇಟ್ ಕಾಪಿ ಆದಾಗ ಏನಾಗುತ್ತೆ? ಇದು ಆಕ್ಚುಲಿ ಈ ತರ ಕಾಪಿ ಆಗುತ್ತೆ. ಇಲ್ಲಿ ಈ ತರ ಇದೆಲ್ಲ 2 ಪಾರ್ಟ್ ಆಗುತ್ತೆ. ಇಲ್ಲೇ ನೆನಪಿರಲಿ, ಸುಮ್ಮನೆ ಒಂದು ಕಾಪಿ ಮಾಡ್ಬಿಟ್ಟು ಇನ್ನೊಂದನ್ನ ಪುಷ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ ಆಚೆ. ಯಾಕೆ? ಇಫ್ ಯು ಪುಷ್ ಔಟ್ ಅದರ್ DNA ಔಟ್ ಆಫ್ ದ ಸೆಲ್, ಆ DNA ಸರ್ವೈವ್ ಆಗಲ್ಲ. ಸೊ ಒಂದು ಸಪ್ರೇಟ್ ಮನೆ ಮಾಡ್ಕೊಡ್ಬೇಕು ಅದಕ್ಕೂ. ದಟ್ ಇಸ್ ಸೆಲ್ ಅಪ್ಯಾರಟಸ್ ಮಾಡ್ಕೊಡ್ಬೇಕು ಅದಕ್ಕೆ. ಬಿಕಾಸ್ ದ ಕಾಪಿ ಪುಷ್ಡ್ ಔಟ್ ಆಫ್ ವುಡ್ ನಾಟ್ ಹ್ಯಾವ್ ಎನಿ organized cellular structure for the maintenance of life process. ಒಂದು ಸುಮ್ಮನೆ ಆಚೆ ಪುಷ್ ಮಾಡ್ಬಿಟ್ರೆ ಏನಾಗುತ್ತೆ? ಅದಕ್ಕೆ ಲೈಫ್ ಮೇಂಟೈನ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. ಸೊ ಯು ನೀಡ್ ಟು ಮೇಕ್ ಅ ಸಪ್ರೇಟ್ ಹೌಸ್ ಫಾರ್ ಇಟ್. ದಟ್ ಇಸ್ ಅದಕ್ಕೂ ಕೂಡ ನ್ಯೂ ಕಾಪಿ ಇದೆಯಲ್ಲ, ಆ ನ್ಯೂ ಕಾಪಿಗೂ ಕೂಡ ಹೊಸದು ಲೇಯರ್ ಮಾಡ್ಕೊಡ್ಬೇಕು. ದಟ್ ಇಸ್ ಸೆಲ್ ಅಪ್ಯಾರಟಸ್ ಮಾಡ್ಕೊಡ್ಬೇಕು. ದಟ್ ಇಸ್ therefore the DNA copy is accompanied by creation of additional cellular apparatus. So DNA copy ಆಗೋದು ಮಾತ್ರ ಅಲ್ಲ, when DNA is getting copied, even the cell apparatus needs to be get copied or needs to be replicated. ಆ cell apparatus ಕೂಡ ಸಪ್ರೇಟ್ ಆಗಿ ಫಾರ್ಮ್ ಆಗಬೇಕು. And then the DNA copies separate, each with its own cellular apparatus. Effectively, a cell divides to give rise to a 2 cells. So one cell in the ಎರಡು cell copy ಆಗಬೇಕಾದ್ರೆ DNA is getting copied and also the cell apparatus is also getting copied. Now these two cells are of course similar, but they are likely to be absolutely identical. DNA copy ಆಗಬೇಕಾದ್ರೆ are they identical? Obviously not. ಈ DNA copy ಆಗಬೇಕಾದ್ರೆ there would be some small variations. So every single time there is a biochemical reaction, there are certain variations ಆಗುತ್ತೆ, and because of these variations to DNA ಮಧ್ಯದಲ್ಲಿ ಸ್ವಲ್ಪ differences ಬಂದ್ಬಿಡುತ್ತೆ. The answer to this question will depend on how accurately the copying of reaction involves occur. No biochemical reaction is absolutely reliable. That ನಾನು ಹೇಳಿದ್ನಲ್ಲ, ಈ DNA ಕಾಪಿ ಆಗಬೇಕಾದ್ರೆ you cannot be sure that 100% complete 100% ಡಿವೈಡ್ ಆಗಿದೆ ಅಂತ. So there would be some variations, ಸ್ವಲ್ಪ variations ಇದ್ದೇ ಇರುತ್ತೆ. That is, therefore it is only to be expected that the process of copying DNA will have some variations. A DNA copy ಆಗಬೇಕಾದ್ರೆ some variations ಇದ್ದೇ ಇರುತ್ತೆ. The variation each time. As a result, the DNA copy generates will be similar. ಆ DNA ಕಾಪಿ ಆಗಿರುತ್ತಲ್ಲ, it will be similar but not identical ಇರಲ್ಲ. That is, but maybe not identical to the original. Some of these variations might be so drastic. Some of the variations might be so drastic ಅಂದ್ರೆ ಏನು ಅರ್ಥ ಏನಂತಂದ್ರೆ, ಈ variations ಇದೆಯಲ್ಲ, sometimes these variations can be so drastic. ಈ variations ಎಷ್ಟು harmful ಆಗ್ಬಿಡುತ್ತೆ ಅಂದ್ರೆ the cell will not be able to survive, the DNA will not be able to survive. So variations are good and also bad at the same time. So variations are very risky. Some of these variations might be so drastic that the new DNA cannot work with the cellular apparatus, its inheritance. Completely different ಆಗ್ಬಿಟ್ರೆ ಆ cell apparatus ಮಾಡ್ಕೊಟ್ಟಿದೀವಲ್ಲ ಸಪರೇಟ್ ಆಗಿ, ಆ cell apparatus ಎಲ್ಲದು ಸರ್ವೈವ್ ಮಾಡೋಕ್ಕಾಗಲ್ಲ. So it's very important to make sure that when the DNA is copying, ಆ ವೇರಿಯೇಷನ್ ತುಂಬ ಮಿನಿಮಮ್ ಆಗಿರಬೇಕು, ವೇರಿಯೇಷನ್ ತುಂಬ ಮಿನಿಮಮ್ ಇರಬೇಕು, ಆ ಜೊತೆಗೆ ಸೆಲ್ apparatus ಕೂಡ ಕಾಪಿ ಆಗಬೇಕು. Both the factors are very important. Such a newborn cell will simply die. On the other hand, there could still be many other variations in the DNA copy that would not lead to such drastic outcome. Thus, the survival of the cells are similar but subtle differences from each other. So, under artha eno, when DNA is getting copied, each cell is similar, but there would be little, very minute, a fraction of difference irutte. This inbuilt tendency of variation during reproduction ಆಗಬೇಕಾದ್ರೆ variations ಬರ್ತಾ ಇರುತ್ತಲ್ಲ, this could lead to what? Evolution. Is the basis of evolution, as we will discuss in the next chapter. So ಅರ್ಥ ಏನು? For an organism, for a species to evolve, one species evolve ಆಗಬೇಕಂದ್ರೆ variations, the inbuilt variations are extremely important. Inbuilt variations ಆಗಲಿಲ್ಲ ಅಂದ್ರೆ the next generation ಆಗಲ್ಲ. Clear? Evolution kuda aga doubt. So that is the meaning of it. Next paragraph ಏನಪ್ಪ? First, that is importance of variation. ಈ variations ಹೇಳಿದೆ ನಾನು. Variations importance ಏನು? The first importance of variation is evolution. If there is variation happening, evolution will take place. Right? If there is variation, evolution is taking place. Population of organisms fill well-defined places or niches. Niches ಅಂದ್ರೆ ಏನು? Niches ಅಂದ್ರೆ it's a fundamental unit of nature. Niche is the immediate surrounding. ಒಂದು organism the immediate surrounding ಇರುತ್ತಲ್ಲ, and what is its function? ಒಂದು easy example ಕೊಡ್ತೀನಿ ನೋಡಿ. For example, ಒಂದು ಸರೌಂಡಿಂಗ್ ಅಲ್ಲಿ ಒಂದು ಡಂಗ್ ಬೀಟಲ್ ಇದೆ. ಡಂಗ್ ಬೀಟಲ್ ಅಂದ್ರೆ ಒಂದು ಬೀಟಲ್ ಇರುತ್ತೆ which survives in the dung. ಇದು ಕೌ ಡಂಗ್ ಇರ್ಬೋದು, ಕೌ ಡಂಗ್ ಇಲ್ಲ ಅಂದ್ರೆ ಆನಿಮಲ್ ಡಂಗ್ ಅಲ್ಲಿ ಇರುತ್ತೆ ಒಂದು ಡಂಗ್ ಬೀಟಲ್. Its niche is the dung. But a niche ಅಂದ್ರೆ ಏನು? What is the function? What is the job of that dung beetle? ಇನ್ನೊಂದು ಎಕ್ಸಾಂಪಲ್ ತೊಗೊಳ್ಳಿ. ಒಂದು ಚೀತಾ ಅನ್ಕೊಳ್ಳಿ. ಆ ಚೀತಾ is staying in a grassland. ಅದ್ರ ಅಕ್ಕಪಕ್ಕ grassland ಇದೆ, nearby surrounding. Now what is the role of that cheetah? Our job of the cheetah is its niche. So niche is nothing but the immediate surrounding and how the organism is interacting, and what is the role of that organism in its surroundings in the ecosystem, using their ability to reproduce. So a population of organism fill well-defined places. Fit well-defined places or niches in an ecosystem. So ಒಂದು ಪಾಪ್ಯುಲೇಷನ್ಗೆ ಒಂದು ಎಕ್ಸಾಕ್ಟ್ ಲೊಕೇಶನ್, ಒಂದು ಪಾಪ್ಯುಲೇಷನ್ ಆಫ್ ಚೀತಾಸ್ಗೆ ಒಂದು ಗ್ರಾಸ್ಲ್ಯಾಂಡ್ ಫಿಕ್ಸ್ ಆಗಿಬಿಟ್ಟಿದೆ ಮನೆ ಹತ್ರ ಇನ್ ಅನ್ ಎಕೋಸಿಸ್ಟಮ್. And it is using ability to reproduce. Different abilities irutte. The consistency of DNA copying during the reproduction is important for the maintenance of the body design feature that allows the organisms to use that particular niche. Artha enu? ಈ DNA ಕಾಪಿ ಆಗಬೇಕಾದ್ರೆ ವೇರಿಯೇಷನ್ ತುಂಬಾ ಮಿನಿಮಮ್ ಆಗಿರಬೇಕು. Why? If the variations are not minimum, if that on the trait ಇದೆ ನೋಡ್ಕೊಳ್ಳಿ, for example cheetah has a trait of running very fast, a trait follow through ಆಗಿಲ್ಲ ಅಂದ್ರೆ ಅಕಸ್ಮಾತ್ if the cheetah is not able to maintain that trait of running very fast, it becomes very difficult for it to survive in that niche. For example on lion ಅನ್ಕೊಳ್ಳಿ, ಆ ಲಯನ್ಗೆ ಸ್ಟ್ರೆಂತ್ ಇಲ್ಲ ಅನ್ಕೊಳ್ಳಿ, ಆ ನಿಶ್ ಸರ್ವೈವ್ ಆಗುತ್ತಾ? will we still call the lion the king of jungle if he doesn't have the strength, if he doesn't have the roar? ಆಗಲ್ಲ ತಾನೆ. So that it becomes very important that inheritance of trait happens, inheritance of that particular variation happens. That is, consistency of DNA copying during reproduction, consistent copy ಆಗ್ತಿರಬೇಕು, is important for the maintenance of body design. That is, cheetah should be able to run fast. That allows the organisms to use that particular niche. A particular grassland ನ ಯೂಸ್ ಮಾಡಬೇಕಂದ್ರೆ the cheetah should keep on inheriting that body design. A body design ಏನು, a fast running ability ನ inherit ಮಾಡ್ತಾ ಇರಬೇಕು ಅದು. Reproduction is therefore linked to stability of population of species. ಅಕಸ್ಮಾತ್ reproduction ಆಗಲಿಲ್ಲ ಅಂದ್ರೆ stability of population also goes away. Why? Because if there is no reproduction, a transfer of trait ಆಗ್ತಿದ್ಯಾ? ಆಗ್ತಾ ಇಲ್ಲ. Then you might have a cheetah which is not able to run fast, right? If the cheetah is not able to run fast, will it be able to survive in that niche? Absolutely not. So stability of the population will be gone. A stability complete ಆಗಿ ಹೋಗ್ಬಿಡುತ್ತೆ. However, niches can change because of the reasons beyond the control of the organism. ಅರ್ಥ ಏನು? ಆ surroundings ಇದೆಯಲ್ಲ, ಆ surrounding of the organism can completely change. For example, flood ಬಂದ್ಬಿಡ್ತು, forest fire ಆಗೋಯ್ತು, earthquake ಆಗೋಯ್ತು, tsunami ಬಂದ್ಬಿಡ್ತು. All these are natural conditions which is not in control of the organism. ಈ organism control ಮಾಡೋಕ್ಕಾಗಲ್ಲ ಈ different factors ನ. When this happens, the niche changes. Niche change ಆಗ್ಬಿಟ್ಟಿದೆ ಈಗ. Now the organism needs to adapt to it. Akasmat adaptive organism, if the cheetah is able to adapt to the new conditions, it will survive. If the cheetah is not able to adapt to new conditions, it will die. For example, grassland ಅಲ್ಲಿ it is able to hunt now. ಅಕಸ್ಮಾತ್ ಸರ್ಟನ್ ಏನೋ ಒಂದು ಎಲ್ಲ ಪ್ಲಾಂಟ್ಸ್ ಡಿಸೀಸ್ ಬಂದ್ಬಿಡ್ತು. ಈ ಗ್ರಾಸ್ಲ್ಯಾಂಡ್ ಅಲ್ಲಿ ಯೋಚನೆ ಮಾಡಿ, ಆ ಗ್ರಾಸ್ಲ್ಯಾಂಡ್ ಅಲ್ಲಿ ಟಾಲ್ ಶ್ರಬ್ಸೇ ಇಲ್ಲ, ಈಗ ಎಲ್ಲ ಫುಲ್ ಫ್ಲಾಟ್ ಗ್ರಾಸ್ ಆಗ್ಬಿಟ್ಟಿದೆ, ಸ್ವಲ್ಪನೂ ಕವರ್ ಇಲ್ಲ. Will the cheetah will be able to hunt now? No. So cheetah needs to now do what? Cheetah now needs to adapt to the new conditions. ಬೇರೆ ಹಂಟಿಂಗ್ ಮೆಥಡ್ಸ್ ಅನ್ನು ಡೆವಲಪ್ ಮಾಡಬೇಕಾಗುತ್ತೆ ಅದು. ಸೊ ವೆನ್ ದ ಆರ್ಗನಿಸಮ್ ಇಸ್ ಏಬಲ್ ಟು ಅಡಾಪ್ಟ್ ಟು ದ ಡಿಫರೆಂಟ್ ಕಂಡೀಷನ್ಸ್ ಅರೌಂಡ್ ದೆಮ್, ಇಟ್ ವಿಲ್ ಬಿ ಏಬಲ್ ಟು ಸರ್ವೈವ್. Similarly, temperature on earth can go up and down. Water levels can vary, or there could be meteorites hits. That is, meteorite bombs ಗೋದಾಗಬಹುದು ತಾನೆ. To think dinosaurs ಹೋದ್ರಲ್ಲ ಅದು. To think a few example, if a population of reproducing organisms were suited for a particular niche, and if the niche were able to drastically alter, a surroundings ಇದೆಯಾ, surroundings change ಆಗೋಯ್ತು ಈಗ ಒಂದು organismದು, the population could be wiped out. ಅಂದ್ರೆ ಏನು, population, entire population ಕಂಪ್ಲೀಟ್ ಆಗಿ ಡಿಸ್ಟ್ರಾಯ್ ಆಗ್ಬೋದು. ಎಕ್ಸಾಂಪಲ್, ಡೈನೋಸಾರ್ಸ್ ಇರಬೇಕಾದ್ರೆ ಆಯ್ತು, ಕಂಪ್ಲೀಟ್ ಆಗಿ ಮೀಟರಾಯ್ಡ್ ಅಟ್ಯಾಕ್ ಬಂತು, and all the dinosaurs were wiped out. However, if some variations were able to present in the few individuals in these populations, ಅರ್ಥ ಏನು, one of the few individuals ಗೆ ಒಂದು ಪ್ರೊಟೆಕ್ಷನ್ ಸಿಕ್ಬಿಡ್ತು, ವೇರಿಯೇಷನ್ಸ್ ಆಗೋಯ್ತು, DNA ಚೇಂಜ್ ಆಗೋಯ್ತು. And if this, because of this variation, if some individuals are able to survive, the new change, they will give rise to a new species itself. If there would be some change for them to survive, some organisms are able to change themselves for survival. Thus, if there were a population of bacteria, example ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ, for example of bacteria living in a temperate water, and if the water temperature the water temperatures were to be increased by global warming, ಒಂದು ಚಿಕ್ಕದು ವಾಟರ್ ಬಾಡಿ ಇದೆ ಅನ್ಕೊಳ್ಳಿ, ಒಂದು ವಾಟರ್ ಬಾಡಿ ಇದೆ, ಇಲ್ಲಿ ವಾಟರ್ ಬಾಡಿ ಇದೆ, ಈ ವಾಟರ್ ಬಾಡಿ ಏನು ಬೆಳೀತಿದೆ, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬೆಳೀತಿದೆ ನೋಡಿ. A population of bacteria living in a temperate waters, if the water temperature were to increase by global warming. So water body ಅಲ್ಲಿ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇದೆ, ಫುಲ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬೆಳೀತಿದೆ, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಬೆಳೀತಿದೆ ಆರಾಮಾಗಿ. ಈಗ ವಾಟ್ ಇಫ್ ಈ ವಾಟರ್ ಟೆಂಪರೇಚರ್ ಇನ್ಕ್ರೀಸ್ ಆಗೋಯ್ತು, if the water temperature increases because of global warming reason ಇಂದ ಈ ಟೆಂಪರೇಚರ್ ಆಫ್ ವಾಟರ್ ಇನ್ಕ್ರೀಸ್ ಸಡನ್ಲಿ, ಏನಾಗುತ್ತೆ ಅವಾಗ? ಮೋಸ್ಟ್ ಆಫ್ ದೀಸ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ ವುಡ್ ಡೈ. ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಎಲ್ಲ ಸತ್ತೋಗ್ಬಿಡುತ್ತೆ. ಯಾಕೆ? ಬಿಕಾಸ್ ಆಫ್ ದಟ್, ವೆನ್ ದ ಟೆಂಪರೇಚರ್ ಇನ್ಕ್ರೀಸಸ್, ದ ಎಂಜೈಮ್ಸ್ ಇನ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಆರ್ ನಾಟ್ ಏಬಲ್ ಟು process. ಎಂಜೈಮ್ಸ್ ವರ್ಕ್ ಆಗಲಿಲ್ಲ ಅಂತ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇಸ್ ಆಲ್ಸೋ ಡೈಯಿಂಗ್, ಪ್ರೋಟೀನ್ಸ್ ಆರ್ ಗೆಟಿಂಗ್ ಡಿನೇಚರ್ಡ್, ಅಂಡ್ few variant resist to the heat. ಅರ್ಥ ಏನು? ಸಮ್ ಆಫ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ, ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಾಗುತ್ತೆ, ದೇ ಬಿಲ್ಡ್ ಅಪ್ ರೆಸಿಸ್ಟೆನ್ಸ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ನಿಮ್ಗೆ ಗೊತ್ತಾ ಕೋಲ್ಡ್ ಬರುತ್ತೆ, ನಿಮಗೆ ಆಚಿ ಅಂತ ಬರ್ತಾ ಇರುತ್ತೆ ಕೋಲ್ಡ್. do you know there is no cure for cold? ಕೋಲ್ಡ್ ಬರ್ತಾ ಇರುತ್ತೆ, ಹೋಗ್ತಾ ಇರುತ್ತೆ. Why? Because the disease causing for cold, the virus which is causing the cold, exactly, cold barathalla nimge, cold is caused by a virus called as rhinovirus. ಈ ರೈನೋವೈರಸ್ ಇಸ್ ಏಬಲ್ ಟು ಅಡಾಪ್ಟ್ ಎವ್ರಿ ಸಿಂಗಲ್ ಟೈಮ್. ಒಂದು ಮೆಡಿಸಿನ್ ಕೊಡ್ತೀವಿ, ಮೆಡಿಸಿನ್ ಕೊಟ್ಟಿದ ತಕ್ಷಣ ಸರಿ ಹೋಗುತ್ತೆ, ಮತ್ತೆ ಕೋಲ್ಡ್ ಬರುತ್ತೆ ನಿಮಗೆ. ಯಾಕೆ? ಬಿಕಾಸ್ ದ ವೈರಸ್ ಇಸ್ ಏಬಲ್ ಟು ಮ್ಯೂಟೇಟ್. ಆ ವೈರಸ್ ತುಂಬ ಮ್ಯೂಟೇಟ್ ಆಗ್ತಿದೆ ಫಾಸ್ಟ್ ಆಗಿ. ದ್ಯಾಟ್ ಇಸ್ ವೈ ಸ್ಟಿಲ್ ದೇರ್ ಇಸ್ ನೋ ಮೆಡಿಸಿನ್ ಫಾರ್ ಕೋಲ್ಡ್. ಕೋಲ್ಡ್ ಬಂದಾಗೆಲ್ಲ ನಾವು ಆ ಸಿಂಪ್ಟಮ್ಸ್ನ ಸಪ್ರೆಸ್ ಮಾಡ್ತೀವಿ, ಬಟ್ ಕೋಲ್ಡ್ ಹೋಗಲ್ಲ ಅವತ್ತು. ಸಿಮಿಲರ್ಲಿ ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಾಗುತ್ತೆ, ಕೆಲವು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹಿಯರ್ ಸಮ್ ಆಫ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ವಿಲ್ ಬಿಲ್ಡ್ ರೆಸಿಸ್ಟೆನ್ಸ್. ಸಮ್ ಆಫ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ ವಿಲ್ ಬಿಲ್ಡ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಅರೌಂಡ್ ದೆಮ್. Some of the bacteria will build resistance. Temperature increase ಆಗ್ತಿದೆ, ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ರೆಸಿಸ್ಟೆಂಟ್ ಆಗ್ತಿದೆ. ನೋಡಿ, few variant resist to the heat and would survive and grow further. So ಈ variants ಎಲ್ಲ variations ಬಂತು ಕೆಲವರಲ್ಲಿ. Now these bacteria, these variants will be able to survive in the new temperature, and this will become a new species altogether. And thus variation is thus useful for survival of the species over time. So species is variations is very important, because ಇಲ್ಲಿ ನೋಡಿ, ಈ because of the variation, ಈ variation ಇಂದಾನೆ ಏನಾಯ್ತು, ಈ bacteria was able to survive. Similarly throughout the course of evolution, human evolution totally, because of the variations, humans are able to survive. From Homo habilis to Homo erectus to Neanderthal man to Homo sapiens, we have evolved. We have shown variations in the brain size, the cranial capacity change agide. So all these important variations can lead to evolution, and variations are extremely important for a species to survive. I know in detail I explain ಮಾಡಿದೀನಿ ನಾವು, because detailed explanation is needed, right? ನೀವು NCERT ನ ಕಲಿಬೇಕು, ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕು, ಬಹಳ ಮಾಡಬಾರ್ದು. Now we're learning about the different types of asexual reproduction. So let me define you what is asexual reproduction first. Asexual reproduction is a method of reproduction in which asexual reproduction is a method of reproduction in which a single parent is involved. Asexual reproduction. Before we understand types of asexual reproduction, before we understand the types ಕೊಟ್ಟಿದ್ದಾರೆ, let's understand what is asexual reproduction. Asexual reproduction ಅಲ್ಲಿ there is involvement of a single parent. There is involvement of a single parent. What about gametes? There is no fusion of gametes. There is no fusion of gametes. There is no fusion of gametes. Third point, the offspring and then a child. The offspring of the child is genetically, morphologically identical. The offspring ಅಂದ್ರೆ ಏನು? offspring ಅಂದ್ರೆ child. Offspring or child is genetically, morphologically identical to the parents. It is genetically, morphologically here. That is the offspring. Artha aita? Let me clear this for you. Highlight ಹಾಕೊಂಡದ. Now fission ಬಗ್ಗೆ ಕಲಿಯೋದ, right? Fission ಬಗ್ಗೆ ಕಲಿಯೋದ. Aditi is saying yes, I know, I saw the video. Yes, I know, I saw the video. Aditi Gupta, very unfortunate, right? Now we got fission by kalida. Now we learned about what is asexual reproduction by kalsi navu. Asexual reproduction andre ಏನು? Asexual reproduction ಅಂತ ಅಂದ್ರೆ single parent is involved, genetically identical offspring, and also there is no fusion of gametes. Now let's learn about the different types of fission ಬಗ್ಗೆ ಕಲಿಯೋಣ ನಾವು ಈಗ, ಸರಿನಾ? Different types of fission. So first, fission ಅಂದ್ರೆ ಏನಪ್ಪ? Fission ಅಂದ್ರೆ ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಏನು? ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಜಾಯಿನ್ ಆಗೋದು. ಫ್ಯೂಷನ್, FUSSION. ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಏನು? ಜಾಯಿನ್ ಆಗೋದು. ಬರೀತೀನಿ ನೋಡಿ ಇಲ್ಲಿ. ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಏನು? ಫ್ಯೂಷನ್ ಅಂದ್ರೆ ಜಾಯಿನ್ ಆಗೋದು. ಫಿಷನ್ ಅಂದ್ರೆ ಸಪರೇಟ್ ಆಗೋದು. ಫಿಷನ್ FISSION. ಫಿಷನ್ ಅಂದ್ರೆ ಸಪರೇಟ್ ಆಗೋದು. ಫಿಷನ್, ಜಾಯಿನ್ ಆಗೋದು ಫ್ಯೂಷನ್. ಸೊ ಫಿಷನ್ ಬಗ್ಗೆ ಕಲಿಯೋದ ಫಸ್ಟ್, ಫಿಷನ್ ಬಗ್ಗೆ, ಸರಿನಾ? ಫಿಷನ್ ಡಿಫ್ರೆಂಟ್ ಟೈಪ್ಸ್ ಇದೆ. Now for unicellular organisms, ಎಲ್ಲಾಗುತ್ತೆ? Unicellular organisms, cell division or fission leads to creation of new individual. ಅರ್ಥ ಏನು? First line of NCERT is very clear here. It is trying to tell you, in case of unicellular organisms, cell division is equal to reproduction. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಮತ್ತೆ ಓದ್ತೀನಿ ನೋಡಿ. For unicellular organisms like amoeba, ಈ ಸೆಲ್ ಡಿವಿಷನ್ ಇದೆಯಲ್ಲ, ಸೆಲ್ ಡಿವಿಷನ್ ಆರ್ ಫಿಷನ್ ಲೀಡ್ಸ್ ಟು ಕ್ರಿಯೇಷನ್ ಆಫ್ ನ್ಯೂ ಇಂಡಿವಿಜುವಲ್. ಕ್ರಿಯೇಷನ್ ಆಫ್ ನ್ಯೂ ಇಂಡಿವಿಜುವಲ್ ಅಂದ್ರೆ ಏನು? ಕ್ರಿಯೇಷನ್ ಆಫ್ ನ್ಯೂ ಇಂಡಿವಿಜುವಲ್ ಅಂದ್ರೆ ರೀಪ್ರೊಡಕ್ಷನ್. So cell division is equal to what? Cell division is equal to reproduction in unicellular organisms. First line, very important. Many different patterns of fission have been observed. Different patterns in the no, it can happen in many planes. Any plane, longitudinal, horizontal, right? Different types of fission. Many bacteria and protozoa simply split into two equal halves during the cell division. In organisms such as amoeba, the splitting of two cells during the division can take place in any plane. This is the most important line here. See, nodi, it says amoeba can divide in what? In any plane. Andre artha eno? Nodi illi. Correct kelskoli. So amoeba can divide in any plane. So amoeba can divide in horizontal, longitudinal division agbodu. It can divide longitudinally. Amoeba can divide horizontally. Amoeba can divide like this. Young can divide agbodu amoeba. So amoeba can divide in any plane. Very important word. Why? Because Leishmania can divide only in the longitudinal. Only in vertical division. Vertical division is seen in Leishmania. Clear? Now, so amoeba splits into two cells during the division and can take place in any plane. So cell division can take place in any plane in the case of amoeba, but only longitudinal division, only vertical division in the case of Leishmania. Now let me quickly explain you the method here. Correct explain martini. Now this is one cell. This is the adult amoeba. Adult amoeba is there. Now first step here is duplication of DNA. The first step is duplication of DNA. Duplication of DNA. Second step is what? Duplication of nucleus. Nucleus divide ಆಗ್ತಿದೆ. Division of nucleus, that's called as chirokinesis. Division of nucleus. Then third step ಅಲ್ಲಿ ನೋಡಿ. Third step ಏನಾಗ್ತಿದೆ? Cytoplasm divide ಆಗ್ತಿದೆ. Because when we learned duplication of DNA ಕಲ್ತ್ನವಲ್ಲ, there should be a new cell apparatus also. Cell apparatus is important because DNA cannot survive on its own. A cell apparatus ಬೇಕು. Hence, third step ಅಲ್ಲಿ cytokinesis ಆಗ್ತಿದೆ. That is division of cytoplasm, and finally we have the two daughter cells. However, next step. However, some unicellular organisms show somewhat more organization of their bodies, such as seen in Leishmania, which is a causative agent of Kala-Azar. This is a one-mark question. This is a famous one-mark question, which have a whip-like structure at one end. For motility, in such organisms, binary fission occurs in a definite orientation in relation to these structures. So definite orientation andre no, the binary fission can be seen here in the longitudinal section. Binary fission will happen like this. But in case of amoeba it can happen in any plane. But in the case of Leishmania, it is vertical. Agi divide agutte Leishmania into two different parts. Clear? Next. Now that is the two type of fission. Fissionally, normal fission, binary fission, we have two types. Binary fission which can happen in any plane. Amoeba example. Binary fission which can happen in only one plane, proper orientation, Leishmania. Now, on other single cell organisms such as Malaria's parasite. Remember, malaria is caused by a parasite. Malaria is not caused by mosquitoes. Malaria is not caused by mosquitoes. Mosquitoes are the carrier of malaria. Very important line. So the causative agent of malaria is Plasmodium. Plasmodium is the causative agent. Plasmodium divides into many daughter cells simultaneously by multiple fission. Example of multiple fission, ನಿಮ್ಮ NCERT ಅಲ್ಲಿ ಕೊಟ್ಟಿರೋದೇನು? Plasmodium. Very important. One mark question, direct ಕೇಳ್ತಾರೆ. ನಿಮ್ಮ example ಬರ್ಬೋದು. The example of multiple fission ಅಲ್ಲಿ ನಿಮ್ಗೆ ಕೊಡ್ತಾರೆ. Examples ಕೊಡ್ಬೋದು, Leishmania ಕೊಡ್ಬೋದು, Plasmodium ಕೊಡ್ಬೋದು, amoeba. But remember, amoeba also does multiple fission. But ನಿಮ್ಮ NCERT ಅದು ಕೊಟ್ಟಿಲ್ಲ. So please learn the example which is given in your NCERT, that is Plasmodium is the example of multiple fission. Yeast, on the other hand, can put up small buds that separate and grow further as we see in the activity 7.1. So budding can be seen in case of yeast. So budding is a separate topic, but they are talking about hydra ಬಗ್ಗೆ ಮಾತಾಡ್ತಾರಲ್ಲಿ. They'll talk about hydra, but you should remember budding can also be seen where? Budding can also be seen in yeast. Next one, fragmentation. So first one done. fission ಆಯ್ತು. Fission ಅಲ್ಲಿ we have two types, binary fission, multiple fission. Binary fission ಅಲ್ಲಿ two types ಇದೆ. Binary fission which can happen in any plane. Example is amoeba. Binary fission which is happening in only one plane. Example ಏನಪ್ಪ ಅದು? Example is leishmania. Then we have multiple fission. Multiple fission example ಏನು? Plasmodium. So all these examples are very important for 1 mark question. Now next one is fragmentation. ಫ್ರಾಗ್ಮೆಂಟೇಷನ್ ಅಂದ್ರೆ ಏನು? ಡಿಫ್ರೆಂಟ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್. ಒಂದು ಮಿರರ್ ತೊಗೊಂಡು ಗ್ಲಾಸ್ನ ತೊಗೊಂಡು ಹೊಡೆದಾಕಿತ್ತಾಳೆ, ಏನಾಗುತ್ತೆ? ಡಿಫ್ರೆಂಟ್ ಫ್ರಾಗ್ಮೆಂಟ್ಸ್ ಆಗ್ಬಿಡುತ್ತೆ, ಗ್ಲಾಸ್ ಪೀಸಸ್ ಆಗ್ಬಿಡುತ್ತೆ. ಒಂದು ದೊಡ್ಡ ಗ್ಲಾಸ್ ಪೀಸಸ್ ಆಗೋಗುತ್ತೆ. ಸೇಮ್ ಈ ಫ್ರಾಗ್ಮೆಂಟೇಷನ್ ಅದೇ ಆಗೋದು. ಒಂದು ದೊಡ್ಡ ಆರ್ಗನಿಸಮ್ ಇರುತ್ತೆ, ಆರ್ಗನಿಸಮ್ ಏನಾಗುತ್ತೆ? ಸ್ಪ್ಲಿಟ್ ಆಗಿ ಡಿಫ್ರೆಂಟ್ ಪಾರ್ಟ್ಸ್ ಆಗ್ಬಿಡುತ್ತೆ ನೋಡಿ. ಇನ್ನು ಮಲ್ಟಿ ಸೆಲ್ಯುಲಾರ್, ಇಲ್ಲಿವರೆಗೂ ನಾವು ಯೂನಿಸೆಲ್ಯುಲಾರ್ ಮಾತಾಡ್ತೀವಿ. ಯೂನಿಸೆಲ್ಯುಲಾರ್ ಈಗ ಫ್ರಾಗ್ಮೆಂಟೇಷನ್ ಹ್ಯಾಪನಿಂಗ್ ಇನ್ multicellular organisms with relatively simple body organization. Simply reproductively methods can be still work. That is, in Spirogyra for example simply breaks up into pieces upon maturation. Spirogyra ಏನಾಗುತ್ತೆ? Spirogyra upon maturation, Spirogyra mature ಆಗಿದ ತಕ್ಷಣ ಏನಾಗುತ್ತೆ? ಪೀಸಸ್ ಆಗಿ ಮುರಿದೋಗ್ಬಿಡುತ್ತೆ. These pieces or fragments grow into a new individual. Each single piece ಇದೆಯಾ ನೋಡಿ, ಅಕಸ್ಮಾತ್ ಒಂದು ಗ್ಲಾಸ್ ಪೀಸ್ ಅದನ್ನ ಹೊಡೆದಾಕಿದ್ರಿ ನೀವು, ಗ್ಲಾಸ್ ಪೀಸ್ ಹೊಡೆದ ತಕ್ಷಣ ಒಂದೊಂದು ಚಿಕ್ಕ ಪೀಸ್ ಬಂತು. ನಾವು ಇಮ್ಯಾಜಿನ್ ಮಾಡಿ ಆ ಚಿಕ್ಕ ಪೀಸು ಕೂಡ ಒಂದೊಂದು ಪೀಸು ಕೂಡ ಕಂಪ್ಲೀಟ್ ಆಗಿ ಒಂದು ಫುಲ್ ಗ್ಲಾಸ್ ಆಗ್ಬಿಟ್ರೆ ಹೆಂಗಿರುತ್ತೆ? That is happening in Spirogyra. That is one single mature Spirogyra is breaking into many pieces, and each piece is able to grow into a completely new adult. Now this is not true for all multicellular organisms. They cannot simply divide by cell. For example, we humans cannot divide cell by cell, right? The reason is that many multicellular organisms, as we have seen, are not simply a random collection of cells. We are not a random collection of cells. We have specialized cells. We have specialized cells. For example, in the plants we have what? In the plants we have xylem, phloem, parenchyma, collenchyma, sclerenchyma. Not just random cells. We have cells for a specific function even in the plants, right? Specialized cells are organized as tissues. Group of cells is known as what? Group of cells is known as tissues, and tissues are organized into organs which they have been placed at a definite position in the body in such a carefully organized situation. Artha ಏನು? Cell organization ಇದೆ ಬಾಡಿಯಲ್ಲಿ. ಅರ್ಥ ಏನು? ಇನ್ನೊಂದು ಅರ್ಥ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Each and every cell has a function. Each and every tissue has a function in our body. So we cannot randomly divide all the cells. Right? We cannot randomly divide all the cells. In a such of carefully organized situation, cell by cell division would be impractical. Multicellular organisms therefore need to use more complex ways of reproduction. So in the case of multicellular organisms, there needs to be a complex level of reproduction ಆಗುತ್ತೆ. For example ಏನಪ್ಪಾ, a basic strategy is used in multicellular organisms is that different cell types ಹೇಳಿದ್ನಲ್ಲ, case of plants, xylem, phloem, parenchyma, collenchyma, sclerenchyma, smooth muscle cells, right? ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ ಸ್ಮೂತ್ ಮಸಲ್ ಸೆಲ್ಸ್ ಇದೆ, ಬೇರೆ ಟೈಪ್ಸ್ ಆಫ್ ಟಿಶ್ಯೂಸ್ ಇದೆ ನಮ್ಮ ಬಾಡಿಯಲ್ಲಿ, ಸ್ಮೂತ್ ಟಿಶ್ಯೂಸ್, right? So is that different cell types perform different specialized functions. So each and every cell type ಗೆ different function ಇರುತ್ತೆ. White blood cell ಆಗಿರ್ಬೋದು, red blood cell ಆಗಿರ್ಬೋದು. So each and every cell, right? Blood cells transportation of oxygen, white blood cell fighting infection. So each and every cell has a definite function in the body. In the case of multicellular, ನಮ್ಮ human body multicellular. So in the case of multicellular, every single cell has a definite function. Now following this general pattern, reproduction is such an organism is also function of a specific cell type. So reproduction kuda one specific cell irutte, body allu kuda one specific cell irutte which will take care of reproduction. How is this reproduction to be achieved from a single cell type if the organism itself consists of many cell types? The answer is that there must be a single cell type in the organism that is capable of growing, proliferating and making other cell types under the right circumstances. Artha ಏನು? We have one single cell type, such special cell type which can later proliferate. ಅಂದ್ರೆ ಏನು? It can grow, ಬೆಳೆಯುತ್ತೆ ಅದು ಸೆಲ್ಲು, ಬೆಳೆದ್ಬಿಟ್ಟು it can divide and it can form all the different type of cells. That is called as totipotent cells, meristematic cells, stem cells in the humans. So all these are special type of cells which can give rise to a brand new type of cell. Now regeneration ಬಗ್ಗೆ ಓದೋದಾ? regeneration. One thing which you need to know is the difference between regeneration and fragmentation. Still so many students do not know the difference between regeneration and fragmentation. Please learn the difference between regeneration and fragmentation. Nimge innೂ ಗೊತ್ತಿಲ್ಲ ಅಂದ್ರೆ please go watch my detailed video on this chapter where I have discussed complete the difference between regeneration and fragmentation. Now, many fully differentiated organisms have ability to grow, ability to give rise to a new individual organism from their body parts. That is, if an individual is somehow cut or broken into pieces, many of these pieces grow into separate individuals. Many of this, each one is able to grow into a separate individual. For example, simple animals like ಹೈಡ್ರಾದಲ್ಲೂ ಆಗುತ್ತೆ. ಈಚ್ ಎವ್ರಿ ಪಾರ್ಟ್ ಆಫ್ ಹೈಡ್ರಾ, ಒಂದು ಟೆಂಟಿಕಲ್ ಕಟ್ ಆಯ್ತು ಅನ್ಕೊಳ್ಳಿ, ಒಂದು ಟೆಂಟಿಕಲ್ ಇಂದ ಕಂಪ್ಲೀಟ್ ಬಾಡಿ ಡಿಸೈನ್ ಆಗ್ಬೋದು. ಒಂದು ಸಿಂಗಲ್ ಪಾರ್ಟ್ ಆಫ್ ಹೈಡ್ರಾ ಕಟ್ ಆಗಿತ್ತು, ಆ ಸಿಂಗಲ್ ಕಟ್ ಆಗಿರೋ ಪಾರ್ಟ್ ಇಂದ ಒಂದು ನ್ಯೂ ಆರ್ಗನಿಸಮ್ ಬೆಳಿಬೋದು, ರೈಟ್? Hydra and planaria can be cut into many number of pieces and each piece grows into a complete organism. This is known as regeneration. ಈ ಪ್ರಾಸೆಸ್ ನ ಕರಿತೀವಿ regeneration. regeneration ಅಂದ್ರೆ ಏನು? ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಆರ್ಗನಿಸಮ್ ಇದೆ, ಪ್ಲನೇರಿಯಾ. ಆ ಪ್ಲನೇರಿಯಾ ಏನಾಯ್ತು, ಮಿಸ್ಟೇಕ್ ಆಗಿ ಏನೊಂದು ನ್ಯಾಚುರಲ್ ಫಿನೋಮಿನಾ ಆಗಿ ಕಟ್ ಆಗೋಯ್ತು ಅನ್ಕೊಳ್ಳಿ, ಪೀಸ್ ಆಗೋಯ್ತು. Now each piece is able to grow into a regenerate into a completely new organism. Each every single piece is able to grow into a completely new organism. Now regeneration is carried out by, regeneration process ideyala, it is carried out by a specialized cells. These cells proliferate ಅಂದ್ರೆ no, they grow and make a large number of cells. These special cells are able to grow and make a lot of cells. Right? They are able to make a lot of cells. From this mass of cells, different cell undergo changes to become various cell types and tissues. So ಒಂದು ಸೆಲ್ ಇದೆ, ಡಿವೈಡ್ ಆಯಿತು, ಡಿವೈಡ್ ಆಗಿ ಒಂದು ದೊಡ್ಡ ಮಾಸ್ ಆಫ್ ಸೆಲ್ ಬಂದ್ಬಿಡ್ತು. ಈ ಮಾಸ್ ಆಫ್ ಸೆಲ್ ಇದೆಯಲ್ಲ, ಈ ಮಾಸ್ ಆಫ್ ಸೆಲ್ ಇಸ್ ಏಬಲ್ ಟು ನೌ ಸಪ್ರೇಟ್ ಔಟ್ ಇಂಟು ಅ ಸ್ಪೆಷಲ್ ಡಿಫರೆಂಟ್ ಟೈಪ್ ಆಫ್ ಸೆಲ್ಸ್. ನೋಡಿ, from this mass of cells, different cells undergo change and become various cell types and tissues. So mass of cells you will get what? From the mass of cells, you will have different types of cell formation. Now, these changes takes place in an organized sequence referred to as development. However, very important line, however, regeneration is not the same as reproduction. Regeneration is not the same as reproduction because why? Because since most of the organisms would not normally depend upon being cut in able to reproduce. So these organisms cannot wait just until they are cut. Until they are cut, they are not able to form a new species. So organisms cannot wait until they are cut. So reproduction is very different here, right? So reproduction is not same as regeneration. Clear agirbeku point 2. Next point, budding. Next one topic is budding. Budding is seen in where? Budding is seen in the case of hydra as well as yeast. Remember the examples ಕೇಳ್ತಾರೆ. ಈ ಒನ್ ಮಾರ್ಕ್ ಕ್ವೆಶ್ಚನ್ ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ ನಿಮಗೆ, ವಾಟ್ ಇಸ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್ ಆಫ್ ಈಸ್ಟ್, ವಾಟ್ ಆರ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್ ಆಫ್ ಬಡಿಂಗ್? ಹೈಡ್ರಾ ಆಗಿರಲಿ, ಈಸ್ಟ್ ಆಗಿರಲಿ, ದ 2 examples which is given in your NCERT ನಿಮಗೆ ನೆನಪು ಇರಬೇಕು ಅದು. Now, budding ಬಗ್ಗೆ ಕಲಿಯೋಣ. Organisms such as hydra use regenerative cells for reproduction in the process of budding. ಒಂದ್ಸತಿ ಹೇಳ್ತೀನಿ, ಕರೆಕ್ಟಾಗಿ ಕೇಳಿಸ್ಕೊಂಬಿಡಿ ನೋಡಿ ಇಲ್ಲಿ. ಸೊ ಇದು ಹೈಡ್ರಾ ಬಡ್ ಇದೆಯಲ್ಲ, ಇದು ಸ್ಮಾಲ್ ಔಟ್ಗ್ರೋತ್ ಆಗುತ್ತೆ. ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲಿ ಒಂದು ಸ್ಮಾಲ್ ಔಟ್ಗ್ರೋತ್ ಬರುತ್ತೆ. ಈ ಸ್ಮಾಲ್ ಔಟ್ಗ್ರೋತ್ ಇಂದ ಆನ್ ದಿಸ್ ಸೈಡ್, ಈ ಸೈಡ್ ಇದೆಯಲ್ಲ, ದೇರ್ ವಿಲ್ ಬಿ ರಿಪೀಟೆಡ್ ಸೆಲ್ ಡಿವಿಷನ್ ಆಗುತ್ತೆ. ಕಂಪ್ಲೀಟ್ ಆಗಿ ಸೆಲ್ ಡಿವಿಷನ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅಂಡ್ ಬಿಕಾಸ್ ದ ರಿಪೀಟೆಡ್ ಸೆಲ್ ಡಿವಿಷನ್, ಆ ಸ್ಮಾಲ್ ಬರ್ಡ್ ಬರುತ್ತೆ. ದಿಸ್ ಸ್ಮಾಲ್ ಬರ್ಡ್ ವಿಲ್ ಡಿರೈವ್ ದ ನ್ಯೂಟ್ರಿಷನ್ಸ್, ಕಂಪ್ಲೀಟ್ ಆಗಿ ನ್ಯೂಟ್ರಿಷನ್ಸ್ ನಾವು ತಗೊಳುತ್ತೆ. ಎಲ್ಲಿಂದ? ದಿಸ್ ಸ್ಮಾಲ್ ಬರ್ಡ್ ವಿಲ್ ಟೇಕ್ ದ ನ್ಯೂಟ್ರಿಷನ್ಸ್ ಫ್ರಮ್ ದ ಪೇರೆಂಟ್ ಹೈಡ್ರಾ. ಇದು ಪೇರೆಂಟ್ ಹೈಡ್ರಾ, ಕಂಪ್ಲೀಟ್ ಆಗಿ ನ್ಯೂಟ್ರಿಷನ್ಸ್ ಪೇರೆಂಟ್ ಹೈಡ್ರಾನ ಎಳ್ಕೊಳುತ್ತೆ ನ್ಯೂಟ್ರಿಷನ್ಸ್ನ. ಅಂಡ್ ಫೈನಲಿ ದಿಸ್ ಸ್ಮಾಲ್ ಬರ್ಡ್ ದೊಡ್ಡದಾಗಿ ಬೆಳೆದ್ಬಿಟ್ಟು ಇಟ್ ವಿಲ್ ಕಂಪ್ಲೀಟ್ಲಿ ಡಿಟ್ಯಾಚ್ ಇಟ್ ಸೆಲ್ಫ್, ಕಂಪ್ಲೀಟ್ ಆಗಿ ಡಿಟ್ಯಾಚ್ ಆಗಿ ಸಪ್ರೇಟ್ ಆಗಿ ನ್ಯೂ ಹೈಡ್ರಾ ಆಗ್ಬಿಡುತ್ತೆ. ಅದೇ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ ಇಲ್ಲಿ, ಅದೇ ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ ಇಲ್ಲಿ. In hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. ಈ ಒಂದು specific site ಅಲ್ಲಿ ಏನಾಗ್ತಿದೆ? repeated cell division ಆಗ್ತಿದೆ. ಈ particular site ಅಲ್ಲಿ repeated cell division ಆಗ್ತಿದೆ. Now these bud develop into a tiny individuals and when fully mature they detach from the parent body and become a new independent individuals. Finally detach ಆಗ್ಬಿಟ್ಟು new individual create ಆಗೋಗುತ್ತೆ. Now next one, vegetative propagation, very important because ಈ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್ ಕಾನ್ಸೆಪ್ಟ್ ಕೇಳ್ತಾರೆ ಯಾವಾಗೂ. ಒಂದು ಕೇಸ್ ಬೇಸ್ಡ್ ಸ್ಟಡಿ ಆಗಿ ಬರ್ಬೋದು, ಇದು ಕಂಪ್ಲೀಟ್ ಕೇಸ್ ಬೇಸ್ಡ್ ಸ್ಟಡಿ ಬರ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ ಒಂದು ಡೈರೆಕ್ಟ್ ಕ್ವಶನ್ ಬರ್ಬೋದು. ವಾಟ್ ಆರ್ ದ ಅಡ್ವಾಂಟೇಜಸ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್, ರೈಟ್? ವಾಟ್ ಆರ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್? ಸಿಂಪಲ್. ವಾಟ್ ಆರ್ ದ ಡಿಸ್ಅಡ್ವಾಂಟೇಜಸ್? ವಾಟ್ ಆರ್ ದ ಎಕ್ಸಾಂಪಲ್ಸ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್? ಎಲ್ಲ ಕೇಳ್ಬೋದು ನಿಮಗೆ. Now, what is vegetative propagation? Kaliyada. Now, there are many plants in which parts like root, stem and leaves, root, stem and leaf develop into new plants under appropriate conditions. So, artha eno? When a plant is growing from other than the seed, and now seed ಆಗ್ತಲ್ಲ, usually planting grow ಆಗುತ್ತೆ. usually plant grow ಆಗೋದು ಹೆಂಗೆ? ನೀವು ಒಂದು ಸೀಡ್ ತಗೋತೀರಾ, ಸೀಡ್ ತಗೋತೀರಾ, ಒಂದು ಬೀಜ ತಗೋತೀರಾ, ಬೀಜ ತೊಗೊಂಡ್ಬಿಟ್ಟು ನೆಲದ ಮೇಲೆ ಹಾಕ್ತೀರಾ, ಮಣ್ಣಲ್ಲಿ ಹಾಕ್ತೀರಾ, ಮಣ್ಣಲ್ಲಿ ಮುಚ್ತೀರಾ, ಆಮೇಲೆ ಮಣ್ಣಲ್ಲಿ ಮುಚ್ಚಾದ್ಮೇಲೆ ಪ್ಲಾಂಟ್ ಗ್ರೋ ಸ್ಟಾರ್ಟ್ ಆಗುತ್ತೆ. ಬಟ್ ಇನ್ ದ ಕೇಸ್ ಆಫ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್ ನೀವು ಸೀಡ್ಸ್ ಯೂಸ್ ಮಾಡಲ್ಲ. ಯು ವಿಲ್ ಯೂಸ್ ಪ್ಲಾಂಟ್ ಪಾರ್ಟ್ಸ್. ಆ ಪ್ಲಾಂಟ್ದು ಒಂದು ಪಾರ್ಟ್ಸ್ ಯೂಸ್ ಮಾಡ್ತೀರಾ, ರೂಟ್ ಆಗಿರಲಿ, ಸ್ಟೆಮ್ ಆಗಿರಲಿ, ಮೆರಿಸ್ಟಮ್ ಆಗಿರಲಿ. ಸೊ ಎವ್ರಿ ಪಾರ್ಟ್ ಆಫ್ ಅ ಪ್ಲಾಂಟ್ ಕ್ಯಾನ್ ಬಿ ಯೂಸ್ಡ್ ಆ್ಯಂಡ್ ಮೇಡ್ ಇನ್ ಟು ಅ ನ್ಯೂ plant. Unlike in most of the animals, plants can indeed use such mode of reproduction. So this is ability of plants. Why? Because plant cells are what? Totipotent cells. They're what? Totipotent cells. This property of vegetative propagation is used in methods such as layering or grafting. Layering and grafting are artificial methods of propagation. Layering, jasmine ಅಲ್ಲಿ ನೋಡಿರ್ತೀರಾ, ಒಂದು ದೊಡ್ಡ ಲೇಯರಿಂಗ್ ಇರುತ್ತೆ, ಜಾಸ್ಮಿನ್ ಇರುತ್ತೆ, ತಡೆ ಹಾಕ್ತೀರಾ ಆಗುತ್ತೆ. ಗ್ರಾಫ್ಟಿಂಗ್ ಅಂದ್ರೆ ಒಂದು ಸೈಕಾನ್ ಮತ್ತೆ ಒಂದು ಸ್ಟಾಕ್ ಇರುತ್ತೆ, ದಟ್ ಇಸ್ ಗ್ರಾಫ್ಟಿಂಗ್. to grow many plants like sugarcane, roses, grapes for agriculture purposes. ಒಂದು ನಿಮಿಷ. Now, advantages. ಇಲ್ಲಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, ಕಡೆ ಕೇಳಿಸ್ಕೊಳ್ಳಿ ಈಗ. ಅಡ್ವಾಂಟೇಜ್: plants raised by vegetative propagation can bear flowers, fruits earlier than the produced from the seeds. First advantage ಬರ್ಕೊಳ್ಳಿ. ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ಲಿ ಹಿಂಗೆ ಮಾರ್ಕ್ ಮಾಡ್ಕೊಂಬಿಡಿ ನೀವು. ಫಸ್ಟ್ ಅಡ್ವಾಂಟೇಜ್ ಪೆನ್ ಚೇಂಜ್ ಮಾಡೋದ, ಒಂದೇ ನಿಮಿಷ ನೋಡಿ ಇಲ್ಲಿ. ಫಸ್ಟ್ ಅಡ್ವಾಂಟೇಜ್ ಏನಪ್ಪ? ಫಸ್ಟ್ ಅಡ್ವಾಂಟೇಜ್ ಏನು? ಪ್ಲಾಂಟ್ಸ್ ರೈಸ್ಡ್ ಬೈ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್ ಕ್ಯಾನ್ ಬೇರ್ ಅಂದ್ರೆ ಇಟ್ ಕ್ಯಾನ್ ಪ್ರೊಡ್ಯೂಸ್ ಫ್ಲವರ್ಸ್ ಮತ್ತೆ ಫ್ರೂಟ್ಸ್ ತುಂಬಾ ಬೇಗ ಬರುತ್ತೆ. ಇನ್ ದ ಕೇಸ್ ಆಫ್ ಹಿಯರ್ vegetative propagation, earlier those produced from the seed. Second one, such methods can also make possible the propagation of plants such as banana, orange, rose and jasmine that has lost the capacity to produce seeds. So we can make plants, we can produce plants from seedless plants. ಈ seedless orange ಇದೆ, seedless banana ಇದೆ, seedless grapes ಇದೆ. ಈ plants ಅಲ್ಲಿ seed ಇಲ್ಲ, ಅದನ್ನ ಹೆಂಗ್ ಬೆಳೆಸ್ತೀರಾ? Vegetative propagation. That is the second advantage. Second advantage growing the plants which do not have the seedless variety. Now, third one, another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parent plants to have its characteristics. Now, why is the third point important? The third point is important. Simple reason. One farmer idane, ಒಂದು banana plant na grow ಮಾಡ್ತಾ ಇದ್ದಾನೆ. ಅಮೇಸಿಂಗ್ ಬನಾನಾಸ್, ತುಂಬಾ ಸ್ವೀಟ್ ಬರ್ತಿದೆ, ದೊಡ್ಡ ಬನಾನಾ ಬರ್ತಿದೆ, ಸ್ವೀಟ್ ಆಗಿರೋ ಬನಾನಾ ಬರ್ತಿದೆ. ಅವ್ನು ಹ್ಯಾಪಿ ಇದ್ದಾನೆ, ಫಾರ್ಮರು, ರೈತ ತುಂಬಾ ಹ್ಯಾಪಿ ಇದ್ದಾನೆ. ನಾವೀಗ ಈ ರೈತಗೆ ಸಪ್ರೇಟ್ ಇನ್ನೊಂದು ಹೊಸ ಲ್ಯಾಂಡ್ ಸಿಕ್ತು ಅನ್ಕೊಳ್ಳಿ, ಹೊಸ ಲ್ಯಾಂಡ್ ಬೈ ಮಾಡ್ದ. ಆ ಲ್ಯಾಂಡ್ ಅಲ್ಲೂ ಕೂಡ ಅವನಿಗೆ ಸೆಕೆಂಡ್ ಫೀಲ್ಡ್ ಅಲ್ಲೂ ಕೂಡ ಸೇಮ್ ಬನಾನಾ ಪ್ಲಾಂಟ್ ಬೇಕು. ಇಸ್ ಇಟ್ ಪಾಸಿಬಲ್? Yes, it is possible. If he does vegetative propagation ಮಾಡಿದ್ರೆ, the banana plant in field A, the banana plant in field A and field B will be completely same. And this is a CBSE 2023 sample paper question. This question which is discussed is from a sample paper. Clear? Similarly, the buds produced in the notches along the leaf margin of bryophyllum. Leaf margin of bryophyllum. Idu nodi illi. Leaf margin of bryophyllum ಅಲ್ಲಿ ಇರುತ್ತೆ, buds ಇರುತ್ತೆ. And when these buds fall on the ground, they will generate a new plant. They'll produce a new plant. Fall in the soil and develop into a new plant. Bryophyllum, example of leaf vegetative propagation. One more example in vegetative propagation, ನೀವು ಪೊಟೆಟೊ ನೋಡಿರ್ತೀರಾ, ಆಲೂಗಡ್ಡೆ ಮೇಲೆ ಏನಿರುತ್ತೆ? ಆಲೂಗಡ್ಡೆ ಮೇಲೆ ಬರ್ಡ್ಸ್ ಇರುತ್ತೆ. ಐ ಆಫ್ ಪೊಟೆಟೊ ಅಂತೀವ, ವೈಟ್ ಪಾರ್ಟ್ ಬರುತ್ತಲ್ಲ, ಆ ವೈಟ್ ಪಾರ್ಟ್ನ ಕಟ್ ಮಾಡಿ ಪೊಟೆಟೊ ಪೀಸ್ ಮಾಡಿಬಿಟ್ಟು ಆ ವೈಟ್ ಪಾರ್ಟ್ ಇರೋದನ್ನ ನೆಲದ ಮೇಲೆ ಹಾಕಿ ಅಲ್ಲಿಂದ ಒಂದು ಪೊಟೆಟೊ ಪ್ಲಾಂಟ್ ಬೆಳೆಸೋಣ ಆಗುತ್ತೆ. ಸೊ ದಟ್ ಇಸ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರೊಪಗೇಷನ್. ನೆಕ್ಸ್ಟ್ ಇಸ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್. ಒಂದು ಸ್ಮಾಲ್ ಟಾಪಿಕ್ ಇದೆ, ಕರೆಕ್ಟಾಗಿ ಮುಗಿಸ್ಕೊಂಡ್ಬಿಡೋದು ಬೇಗ. ಸ್ಮಾಲ್ ಟಾಪಿಕ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್. ಈ ವಿಡಿಯೋ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಟಾಪಿಕ್ ಮೇಲೆ ಒಂದು ಸಪ್ರೇಟ್ ಒಂದು ವಿಡಿಯೋ ಮಾಡಿದ್ದೀನಿ ನಾನು. ಇಟ್ಸ್ ಅ 15 minutes ವಿಡಿಯೋ, ಕಂಪ್ಲೀಟ್ ಪ್ರಾಸೆಸ್ ಆಫ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಅಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ. ಕ್ಲಿಯರ್? ಲೆಟ್ಸ್ ರೀಡ್ ಇಟ್. ಇನ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್, new plants are grown by removing the tissues or separating the cells from the growing tips of the plants. So ನೀವು ಮೆರಿಸ್ಟಮ್ ಪಾರ್ಟ್ ತಗೋಬಹುದು, ಒಂದು ಎಲೆ ತಗೋಬಹುದು, ಒಂದು ರೂಟ್ ತಗೋಬಹುದು, ಒಂದು ಫ್ರೂಟ್ ತಗೋಬಹುದು. So any part of the plant can be taken for a tissue culture and we call it as explant. We call it what? Explant. The cells are then placed in an artificial medium that is called as nutrient medium alaktivi, where they divide rapidly to form a small group of cells or called callus. ಇವು ನ್ಯೂಟ್ರಿಯನ್ ಮೀಡಿಯಂ ಇದೆಲ್ಲ ನ್ಯೂಟ್ರಿಯನ್ ಮೀಡಿಯಮ್ ಅಲ್ಲಿ ಒಂದು ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಇರುತ್ತೆ. ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಅಲ್ಲಿ ನೀವು ಒಂದು ಲಿಕ್ವಿಡ್ ಹಾಕಿರ್ತೀರ, ಆ ಲಿಕ್ವಿಡ್ ಅಲ್ಲಿ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಇರುತ್ತೆ. ಈಗ ನೋಡಿ, ಒಂದು ಪ್ಲಾಂಟ್ಸ್ ಬೆಳೆಸಬೇಕಾದ್ರೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಬೇಕಾನೆ, ಅದಕ್ಕೆ ನೆಲದಿಂದ ಸಾಯಿಲ್ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಇರಬೇಕು. ಫರ್ಟಿಲೈಸರ್ ಹಾಕಿರಲ್ಲ, ಫರ್ಟಿಲೈಸರ್ ಮೆನ್ಯೂರ್. ಸಿಮಿಲರ್ಲಿ ಆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಾ ಇರುತ್ತೆ ಒಂದು ಟೆಸ್ಟ್ ಟ್ಯೂಬ್ ಅಲ್ಲಿ. ಇನ್ ದಟ್ ವಿ ಆಡ್ ಮೈಕ್ರೋ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್, ಮ್ಯಾಕ್ರೋ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಎಲ್ಲ ಆಡ್ ಮಾಡಿರ್ತೀವಿ. ಇನ್ ದಟ್ ವಿ ಆಡ್ ಅ ಪ್ಲಾಂಟ್. ಆ ಪ್ಲಾಂಟ್ ಏನಾಗುತ್ತೆ, ನ್ಯೂಟ್ರಿಯೆಂಟ್ಸ್ ಯೂಸ್ ಮಾಡಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ ಇಂಟು ಅ ಮಾಸ್ ಆಫ್ ಸೆಲ್ಸ್. ಅಂಡ್ ದಿಸ್ ಮಾಸ್ ಆಫ್ ಸೆಲ್ ಇಸ್ ಕಾಲ್ಡ್ as what? This mass of cell is called as callus. It's called as callus. The callus is transferred to another medium containing hormones for growth and differentiation. The plantlets are then placed in a soil. So once we get a new plant, so ಚಿಕ್ಕದಾಗಿ ಒಂದು ಪ್ಲಾಂಟ್ ಬೆಳೆಸಲಾಗುತ್ತೆ, then we transfer it to a soil in a sterile condition. ಆಮೇಲೆ ಅದನ್ನ ಆಚೆ ಕಡೆ ಹಾಕ್ಬೋದು. And this process is called as micropropagation. Grown to mature plants. Using tissue culture, many plants can be grown from one parent in disease-free conditions. This technique is commonly used for ornamental plants. Yeah, ornamental plants because they're expensive, and if you use this technique, cheap agala, but cheap agala. It's a very expensive process, and also we can get disease-free plants for both. So advantage of tissue culture is disease-free plants and also somatic hybrids. What is somatic hybrids? One ಪ್ಲಾಂಟ್ ಕೇಳಿರ್ತೀರ, ಪೊಮ್ಯಾಟೊ ಅಂತ. ಪೊಮ್ಯಾಟೊ ಪ್ಲಾಂಟ್ ಅಂದ್ರೆ ಏನು? ಅದರಲ್ಲಿ ಟೊಮ್ಯಾಟೋನು ಬೆಳೀತಾ ಇರುತ್ತೆ, ಮೇಲೆ ಕೆಳಗಡೆ ಪೊಟ್ಯಾಟೋನು ಬೆಳೀತಾ ಇರುತ್ತೆ. ಸೊ ಸಿಂಗಲ್ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಟೊಮ್ಯಾಟೊ ಮತ್ತು ಪೊಟ್ಯಾಟೊ ಎರಡು ಬೆಳೀತಾ ಇರುತ್ತೆ. ವಿ ಕಾಲ್ ಇಟ್ ಪೊಮ್ಯಾಟೊ ಪ್ಲಾಂಟ್. ಅಂಡ್ ಬಿಕಾಸ್ ಆಫ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಯೂಸ್ ಮಾಡಿಬಿಟ್ಟು, ಬಿಕಾಸ್ ಆಫ್ ಯೂಸಿಂಗ್ ದಿಸ್ ಟೆಕ್ನಿಕ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್, ವಿ ಕ್ಯಾನ್ ಮೇಕ್ ಪೊಮ್ಯಾಟೊ ಪ್ಲಾಂಟ್. ಟೊಮ್ಯಾಟೊ ಅಲ್ಲ, ಪೊಟ್ಯಾಟೊ ಅಲ್ಲ, ಪೊಮ್ಯಾಟೊ. ಸೊ ದಿಸ್ ಇಸ್ ಅ ಪ್ಲಾಂಟ್ ವಿಚ್ ಹ್ಯಾಸ್ ಬೋತ್ ಟೊಮ್ಯಾಟೊ ಅಂಡ್ ಪೊಟ್ಯಾಟೊ ಟುಗೆದರ್. ನೌ ನೆಕ್ಸ್ಟ್ ಒನ್ ಇಸ್ ಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್. ಈ ಸ್ಪೋರ್ ಫಾರ್ಮೇಷನ್ ಅಲ್ಲಿ ಒಂದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಕೊಟ್ಟಿದ್ದಾರೆ ನಿಮಗೆ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ಲಿ. ಒಂದು ಚಿಕ್ಕ ಆಕ್ಟಿವಿಟಿ ಕೊಟ್ಟಿದ್ದಾರೆ. ಒಂದು ಬ್ರೆಡ್ ಪೀಸ್ ತೊಗೊಳ್ಳಿ, ಆ ಬ್ರೆಡ್ ಪೀಸ್ನ ಒಂದು ಅದಕ್ಕೆ ನೀರು ಹಾಕ್ಬಿಟ್ಟು ಒಂದು ರೂಮ್ ಕಾರ್ನರ್ ಇಟ್ಬಿಡಿ, ಡಾರ್ಕ್ ಮಾಯ್ಸ್ಟ್ ಪ್ಲೇಸ್ ಅಲ್ಲಿ. ಆ ಬ್ರೆಡ್ ಪೀಸ್ ಮೇಲೆ ಬೆಳೆಯುತ್ತೆ. ಬ್ರೆಡ್ ಪೀಸ್ ಮೇಲೆ ಒಂದು ವೈಟ್ ಕಲರ್ ಲೇಯರ್ ಬೆಳೀತಾ ಇರುತ್ತೆ. ಅಂಡ್ ದಿಸ್ ವೈಟ್ ಕಲರ್ ಲೇಯರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ವಾಟ್? ದಿಸ್ ವೈಟ್ ಕಲರ್ ಲೇಯರ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಫಂಗಸ್. ಆ ಫಂಗಸ್ದು ಹೈಫೆ. ಇದು ಫಂಗಸ್ ಏನು? ಹೈಫೆ. ಇಲ್ಲಿ ನೋಡಿ, ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ವಾಟ್? ಫಂಗಸ್ ಹೈಫೆ. HYPHAE. ಒಂದು ನಿಮಿಷ ಕ್ಲಿಯರ್ ಬರೆದ್ಬಿಡ. ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ವಾಟ್? ಹೈಫೆ. And group of hyphae is called as what? Mycelium. A group of hyphae is called as mycelium. Even in many simple multicellular organisms, specific reproductive parts can be identified. Reproductive part adu idu sporangiophore ಮತ್ತೆ sporangium. ಇದು ನೋಡಿ, ಇದು sporangium. ಇದು sporangium ಒಳಗಡೆ ಸ್ಪೋರ್ಸ್ ಇರುತ್ತೆ. ಆ ಸ್ಪೋರ್ಸ್ can be liberated. No. A thread-like structure that is developed on the bread in the activity above are hyphae of the bread mold. So, this is hyphae of the bread mold. They are not reproductive parts. Very important. Hyphae is what? Hyphae is vegetative part. What is what? Hyphae is? Hyphae is vegetative part. Hyphae is vegetative part of the fungus. Hyphae is what? Vegetative part of the fungus. Reproductive adu sporangium. Right? On the other hand, the tiny bulb on a stick. Bulb is sporangium, stick is sporangiophore. Right? Structure are involved in reproduction. So this is involved in reproduction. The blobs are sporangia. Idu sporangio. Single is sporangia, which contains cells or spores that can eventually develop into new rhizopus individuals. So ಈ spores ಇದೆಯಲ್ಲ, ಈ spores ಆರೋಗ್ಬಿಟ್ಟು, spores disperse ಆಗ್ಬಿಟ್ಟು, ಎಲ್ಲ every single time the spores travel and there is a favorable conditions ಇದ್ದಾರೆ, ಆಚೆ ಕಡೆ ನೀರಿತ್ತು, ಮಾಯಿಶ್ಚರ್ ಇತ್ತು, ಡಾರ್ಕ್ ಪ್ಲೇಸ್ ಇತ್ತು, each spore will give rise to a new sporangium ಕೊಡುತ್ತೆ ಅದು. Right? The spores are covered by a thick wall that protect them until they become into a contact with another moist surface and begin to grow. So you pour into these spores are extremely resistant irutte, and every single time they find favorable conditions. Only when they find favorable conditions, our spores develop agbiಟ್ಟು new plant produce ಮಾಡುತ್ತೆ. So new fungus produce ಮಾಡುತ್ತೆ. All the modes of reproduction that we have discussed so far allows a next generation to be created from a single individual. Starting early, the definition of asexual reproduction, a single parent is involved, a single individual. This is known as asexual reproduction. That is the complete asexual reproduction. What are the different types of asexual reproduction, different methods of asexual reproduction. So ಇಲ್ಲಿವರೆಗೂ ನೀವು ನೋಡಿದ್ರೆ ನಿಮಗೆ ಅರ್ಥ ಆಯ್ತು. ಅಕಸ್ಮಾತ್ ಇಲ್ಲಿವರೆಗೂ ನಿಮಗೆ ನೋಡಿದಿರಾ ವಿಡಿಯೋ ಅರ್ಥ ಆಗಿದೆ, it was helpful to you, I want all of you to hit the like button and subscribe to the channel right now if you do not want to miss any of the content on this channel, because every single day ಈ ಚಾನಲ್ ಲೈವ್ ಬರ್ತೀನಿ, ಕಂಪ್ಲೀಟ್ ಆಗಿ ನಿಮಗೆ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ, ಎವ್ರಿ ಸಿಂಗಲ್ ಕಾನ್ಸೆಪ್ಟ್ ನ ಲೈನ್ ಆಫ್ NCERT ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ ನಿಮಗೆ. and also you can go watch my long videos where I have explained the chapter in a foundation level, in higher level. So go watch that. Right, now let's learn about sexual reproduction. Let's understand sexual reproduction now. Asexual, done, dusted. Now asexual reproduction start ಮಾಡಕ್ಕಿಂತ ಮುಂಚೆ there are few what do I say, reproductive health ಬಗ್ಗೆ ಹೇಳಿದ್ದಾರೆ. Let's learn that. We are also familiar with the mode of reproduction that depends upon the involvement of two individuals. So ಇಲ್ಲಿ ಬರ್ಬಿಡ್ತೀನಿ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. I'll give you a gist of sexual reproduction. I'll give you a gist of sexual reproduction. ಕರೆಕ್ಟ್ ಕೇಳಿಸ್ಕೊಳ್ಳಿ. Sexual reproduction. There is involvement of, in sexual reproduction, there is involvement of two parents. Involvement of two parents. Next point, in case of sexual reproduction, there is fusion of gametes. Fusion of gametes. Third important point, the offspring here is genetically, morphologically what? Unique. It is genetically, morphologically unique, because in sexual reproduction there is what? Variations ಆಗುತ್ತೆ. ಏನಾಗುತ್ತೆ? Variations ಆಗುತ್ತೆ. Now because of the variations, the offspring is what? Offspring is genetically, morphologically very unique in nature. Now ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳೋಣ. Also before new generation, involvement of two individuals before a new generation can be created. So two individuals. Bulls alone cannot produce a new calf. One bull cannot produce a new calf on its own. Nor can hens alone produce new chicks. Hen get chicken beko. If there are only chicken, then will be able to produce hence chicks. In such cases, both sexes, male and female, are needed to produce a new generations. So there is, in case of sexual reproduction, there is involvement of what? Involvement of two parents, that is both male and female, both male as well as female organs, and both the parents are needed in the case of sexual reproduction. What is the significance of these sexual mode of reproduction? Significance the significance here I'm talking about is variation. Are there any limitations of asexual mode of reproduction? We have discussed below. So now, why the sexual mode of reproduction? Why? What is the need of sexual mode of reproduction? The reason for sexual reproduction, the need of sexual reproduction is what? Variations. Variations create ಆಗುತ್ತೆ. And when there is variations ಅದಕ್ಕೆಲ್ಲ ಏನಾಗುತ್ತೆ? Evolution ಆಗುತ್ತೆ. Organisms will be able to survive, will be in new conditions. Right? The creation of new cells from one individual, copying of DNA, as we have discussed in cellular apparatus. The DNA copying mechanism, as we have noted, cannot be absolutely accurate. DNA copy agಬೇಕಾದ್ರೆ it is not an absolute process. There could be some variations in DNA copy, inbuilt variations, and result in errors as a source of variations in production of new population of organisms. Every individual organism cannot be protected by variations, but in a population, variations are useful for ensuring the survival of species. So I told you, variations ತುಂಬಾ important too. Variations will make sure the species is able to survive. A species it's going to be like, year after year idde irutte a species. So variations are very important. It would therefore make sense if organisms came up with reproductive modes that allow more and more variations to be generated. So if there are more and more variations, useful variations, if there are more and more useful variations, organism will be able to survive more, organism will be able to maintain its life for a longer time. on the face of the earth. That is, one after another generation will be able to be survive. So if there is reproductive modes, if the organism is able to make more reproductive modes, right, if there is more reproduction happening, sexual reproduction is happening, more variations are happening. While DNA copying mechanisms are not absolutely accurate, DNA copying accurate illa, they are precise enough to make generations of variations agbodu. A fairly slow process. So DNA copying mechanism alli variations bartide, but it is a very, very, very slow process adu. If DNA copying mechanisms were be less accurate, many of the resultant DNA copies would not be able to work with the cellular apparatus. That's same munche helidha ಹೇಳ್ತಾರೆ. Each new variation is made up of DNA copy. It already has variations accumulated from the previous generation. So every single time DNA copy ಆಗಬೇಕಾದ್ರೆ ಏನಾಗ್ತಿದೆ ನೋಡಿ, ಇಲ್ಲಿ ಆ ಲೈನ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೊ ಒನ್ DNA ಇದೆ, ಈ ಒಂದು DNA ಇದೆ, DNA ಎರಡು ಕಾಪಿ ಬಂತು, ಈ ಎರಡು ಕಾಪಿ ಏನಾಯ್ತು, ಮತ್ತೆ ಎರಡು ಕಾಪಿ ಬಂತು, ಮತ್ತೆ ಎರಡು ಕಾಪಿ ಬಂತು, ಇದು ಕೂಡ ಎರಡು ಕಾಪಿ ಮಾಡಿತು. So when DNA is copying, ಈ DNA information ಇದೆಯಲ್ಲ, ಈ DNA information ಈ DNA ಇದೆ, ಈ DNA information ಈ DNA ಇದೆ. So whenever the DNA is copying, the information is accumulated. Information is what? Accumulated. ಹೇಳ್ತಾರೆ ನೋಡಿ ಇಲ್ಲಿ. Each new variation is made up of DNA copy that already has variations. ಈ new copy ಇದೆಯಲ್ಲ, ಈ new copy will also have variations from this. This will also have variations from this. So ill variations agidala, the variations happening here, the variations which are happening here will be available here also. Same variations will be available here also. That is, variations accumulated from the previous generations. Our variations are getting accumulated. Thus, two different individuals in a population, two different individuals, individual number 1 A, individual number B here. ಯೋಚನೆ ಮಾಡಿ, ಇಲ್ಲೊಂದು ಬೇರೆ DNA ಇದೆ, ಈ DNA ವೇರಿಯೇಷನ್ಸ್ ಆಗ್ತಿದೆ, ಈ ವೇರಿಯೇಷನ್ ಇಲ್ಲಿ ಎರಡು ಸ್ಪ್ಲಿಟ್ ಆಯ್ತು, ಇಲ್ಲೊಂದು DNA ಇದೆ. ಸೊ ಈ ಪಾಪ್ಯುಲೇಷನ್ they have very different types of DNA. Now what if they reproduce among themselves, right? What if this organism and this organism both are cats? If this cat is reproducing with this cat, the offspring will have what? Variations from this lineage. This will also have variations from this lineage. So complete different lineages in the variations one cat ಬಂದ್ಬಿಡುತ್ತೆ. So whenever there is reproduction, ಅರ್ಥ ಏನು, whenever there is reproduction, lot of variations are getting accumulated. Clear? Whenever there is variation, lot of variations are getting accumulated, right? Since all the variations are in living individuals, it is assured that they do not have any really bad effects iralla. Combining variations from two or more individuals would thus create a new combination of variants. So new variations produce ಆಗ್ತಾ ಹೋಗುತ್ತೆ. Natural ಆಗುತ್ತೆ, but naturally slow ಆಗುತ್ತೆ. Now each combination would be novel and new, since it would involve the two different individuals, and the sexual mode of reproduction incorporates such process of combination of DNA from two different individuals during the reproduction. Artha ಏನು, reproduction ಆಗಬೇಕಾದ್ರೆ there is a mixing of variations is happening from two different individuals in the mixing of variations ಆಗ್ತಿದೆ. But this creates a major difficulty. In each new generation is to be able to a combination of DNA copies of two pre-existing individuals. Then the new generation will end up having twice the amount of DNA. So DNA copy ಆಗಬೇಕಾದ್ರೆ what if there are two copies? ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಇಲ್ಲೊಂದು DNA ಇದೆ, this is from DNA organism number A, this is DNA from organism number D, DNA from organism A, DNA from organism B. If they are able to reproduce, ಇಲ್ಲೊಂದು DNA ಬಂತು, DNA ಇಲ್ಲಿಗ ಡಬಲ್ ಆಗಿಲ್ವಾ ಫುಲ್ಲು, ಇಲ್ಲ 1 2 3 4, ಇಲ್ಲಿ 2 ಇತ್ತು, 1 2 ಬಟ್ ಇಲ್ಲಿ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ ಅಲ್ಲಿ 4 ಬಂದ್ಬಿಡ್ತು. That's what they're talking here. ನೋಡಿ ಇಲ್ಲಿ. But this creates a major difficulty. If each new generation is to be able to if new generation is to be the combination of the new DNA copies. ಹೊಸ generation ಇದೆಯಲ್ಲ, every single new generation is a copy, right? ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಮತ್ತೆ ಹೇಳ್ತೀನಿ, ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, very important line. But this creates a major difficulty. if each new generation is to be the combination of DNA copies from two pre-existing individuals. So individual number A do, individual number DNA, individual number B. If they are procreating, they're making a baby agro, this will have the combination of what? You'll have combination of 4 different DNAs. 1, 2, 3, 4. Right? Each new generation will end up with having twice the amount of DNA that previous generation had. This is likely to mess up the control of the new cellular apparatus by the DNA. How many ways can we think of this showing the more difficulty? So either DNA copy agಬೇಕಾದ್ರೆ, if there is no semi-conservative method, DNA copy agಬೇಕಾದ್ರೆ, there would be 4 times the number of DNA. That would be a major problem. Hence we have semi-conservative method today. Hence we have what? Semi-conservative method of DNA copying. Not really. We have seen earlier that organisms become more complex. So earlier we've seen that the organisms become more complex, the specialization of tissue increases. Right? In the human cell, we have different types of tissues. One solution to that, many multicellular organisms have found for problem mentioned about to have special lineage of cells in specialized organs in which only half the number of chromosomes and half the amount of DNA are compared to the non-reproductive body cells. Artha ಮಾಡ್ಕೊಳ್ಳಿ ಮತ್ತೆ ಇದನ್ನ. This is talking about meiosis here. Meiosis ಬಗ್ಗೆ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಏನು ಹೇಳ್ತಾ ಇದ್ದಾರೆ? Meiosis ಬಗ್ಗೆ. ಒಂದು ನಿಮಿಷ ನೋಡಿ. One solution that many multicellular organisms have found for the problem mentioned above is to have special lineage of cells. Solution ಏನು? Solution is to have special lineage of cells, specialized organs in which only half the number of chromosomes, that is just 23 chromosomes ಇರುತ್ತೆ ಅದರಲ್ಲಿ. In case of humans, we have 46 chromosomes. But in the special cells, example is your sperm. They're talking about sperm here. The sperm is haploid. Sperm is what? Haploid. The sperm will have 23 chromosomes, 23 pair of chromosomes. Right? 23 chromosomes. Right? Only half the amount of DNA as compared to the non-reproducing cells. So we have reproductive cells and we have non-reproductive cells. Clear? Somatic cells and germ cells. This is achieved by the process of cell division called as meiosis. So whenever there is meiosis ಆಗ್ಬೇಕಾದಾಗೆಲ್ಲ, the cell is dividing into from diploid to haploid. So during meiosis 2n to n convert ಆಗ್ತಿದೆ. So when these germ cells, sperm, from the two individuals come together, that is egg in the case of female, sperm in the case of male, during sexual reproduction to form a new individual, it results in a re-establishment of number of chromosomes and the DNA content is in the new generations. So whenever there is reproduction, the haploid gametes meet and which results in a diploid zygote. If the zygote is to grow and develop into an organism which has highly specialized tissues and organs, then it has to have sufficient store energy for doing this. In a very simple organisms, it is seen that two germ cells are not very different from one another. ಅರ್ಥ ಏನು? The two germ cells, ಇಲ್ಲಿ ಮೇಲ್ ಮತ್ತೆ sperm ಇದೆ ನಮ್ದು, sperm ಮತ್ತೆ egg ಇದೆ. They do not look similar. But in the case of some organisms, such as your isogametes in the case of Chlamydia monocelli, isogametes irutte. The male gamete and the female gamete look very alike, or may even be similar. But as the body design becomes more complex, as we become humans, evolution adre, the germ cell also specialize. One germ cell is larger and contain the food store. Example, egg irutte. The egg in the female has storage of food, right? Storage of food very large, while the other is smaller and likely to be motile. Sperm smaller but motile. Conventionally, the motile germ cell is called as male germ cell. Sperm and the female germ cell containing the store food is called as the female gamete. We shall see in the next few sections how the need of to create these two different types of gametes give rise to the differences in the male and female reproductive organs. So based on the germ cell, based on the sperm and the ovum, the reproductive structures are also very different. And in some cases, the differences in the bodies of the male and the female organism. So male and body female organism kuda complete agi ಡಿಫ್ರೆಂಟ್ ಆಗಿರುತ್ತೆ. ಸೊ I'll stop the class here. The next class I'll be talking about sexual reproduction in flowering plants. Then I'll also talk about what? The reproduction in humans. Complete agi cover mada next class alli. So any doubt till now, you can always ask me in the chat. If you do not understand any line, the line artha agilla andre, you can always ask me any line of NCERT. If you do not understand, please ask me. The next class I'll be starting off with the sexual reproduction in flowering plants. Flowering plants kaliyada, human reproduction kuda next class alli kaliyada.